Biochemistry is the quiet science of life at its smallest scale. It is where the language of chemistry merges with the poetry of biology, revealing how molecules move, meet, and transform within every living thing. In its essence, biochemistry studies the subtle choreography of atoms that sustain existence. Every heartbeat, every breath, every blink is guided by countless reactions, too small for the eye to see, yet profound enough to shape every moment of being. As we drift through this calm exploration, imagine yourself shrinking in perspective, descending into the hidden world within your own body, a landscape not of organs and bones, but of molecules in gentle motion. At its heart, biochemistry is the study of matter in motion, not the violent motion of storms or waves, but the rhythmic, almost musical motion of life. Here, water molecules sway and mingle, enzymes fold into delicate shapes, and carbon atoms build endless chains of structure. The field exists at the crossroads of chemistry and biology. Chemistry provides the elements and reactions, while biology gives them meaning and purpose. This is where understanding becomes harmony, where the mechanical becomes living. The word itself, biochemistry, is a fusion of bios, meaning life, and chemistry, the science of matter and change. It reminds us that life is not separate from the physical world, but an intricate part of it, governed by the same natural laws that guide the stars, oceans, and stones. Yet within living systems, these laws manifest in extraordinary ways, combining structure and spontaneity to create what we call vitality. For many, biochemistry can seem abstract or intimidating, but at its core, it is simply the study of how life keeps itself alive, how molecules interact to create warmth, thought, motion, and renewal. All living organisms, from the smallest bacteria to the largest whale, rely on the same molecular language. The same 20 amino acids form proteins in every species. The same sugars feed every cell. The same DNA, with its quiet spiral of information, guides growth and repair. Beneath the diversity of life, there is an elegant unity. Biochemistry uncovers that unity, showing us that the boundary between you, a tree, and a bird is far thinner than it seems. All of us are bound by shared molecules and the same cycles of transformation. When you think of chemistry, you might imagine glass flasks, glowing reactions, or scientists in white coats. But the chemistry of life is gentler, unfolding within cells in perfect quiet. In your body, at this very moment, enzymes are folding and unfolding like silk, binding to other molecules with delicate precision. Sugars are being broken down, energy is being released, and proteins are being synthesized atom by atom. These processes do not stop or sleep. They continue seamlessly, day and night, as though life itself is a soft whisper that never ceases. This subtlety is part of what makes biochemistry so captivating. It reveals that every living being is a vast network of reactions, each dependent on the other, each in balance. A single cell contains thousands of chemical pathways working together in a way that no machine ever could. It is an endless sequence of events where one molecule changes form, releasing energy that fuels another transformation, and so on. Through this, life maintains order in a universe that naturally drifts toward disorder. This constant fight for balance is the foundation of existence, a quiet defiance against chaos. To truly understand biochemistry is to see the beauty in precision. The way molecules recognize each other through shape, how they bond and separate at exactly the right moments, how they form loops of feedback that regulate their own activity. All of it speaks of order, hidden beneath apparent randomness. These molecules are not alive on their own, yet when assembled in the right patterns, they give rise to something that is. It is one of the deepest mysteries in science how inanimate matter gives birth to awareness, motion, and thought. In a way, studying biochemistry is like learning a new form of listening. It trains you to sense patterns that exist beyond the visible. When we speak of reactions, we are really speaking of connections. One molecule hands off an electron to another, energy shifts, and structure follows. This dance of exchange occurs trillions of times every second inside you. It is how cells breathe, how muscles contract, how memory forms. Yet despite all its complexity, it unfolds without sound or sensation, perfectly synchronized, perfectly calm. At the center of this quiet order lies the carbon atom. Carbon is the backbone of biochemistry, a small, versatile element capable of forming 4 bonds at once, linking into chains, rings, and frameworks that define life's architecture. It can bind to oxygen, hydrogen, nitrogen, phosphorus, and sulfur, creating an almost infinite range of molecules. Every protein, lipid, carbohydrate, and nucleic acid you will ever learn about is built upon this element's capacity to connect. In that sense, carbon is the sculptor of life's diversity. Its bonds are strong yet flexible, able to form and reform as needed, making it the foundation of life's adaptability. Water, too, plays an essential role in this story. Life as we know it could not exist without it. Within cells, water forms a supportive environment in which molecules can move and interact. Its polarity allows it to dissolve ions and compounds, making it the perfect solvent for biochemical reactions. When 2 molecules must meet, it is water that carries them together. When reactions must release energy, it is often water that absorbs and redistributes the heat. It is so constant and abundant that we often forget it is a chemical marvel in its own right, a compound whose properties defy simple expectation. As biochemistry studies these materials, it uncovers the principles that connect them: structure and function, energy and balance, reaction and regulation. Every chemical process inside a living being has a purpose to sustain, to repair, to grow, or to adapt. Together, they form networks that scientists call metabolic pathways, where one molecule transforms into another in a series of graceful steps. Even when viewed under the lens of modern science, these pathways retain an almost poetic rhythm. Molecules move like travelers through an invisible landscape, each carrying energy or information to its next destination. But the study of biochemistry is not only about molecules. It is also about the conditions that make their existence possible. Temperature, pH, and concentration must all remain within narrow limits. Too much acidity, too little warmth, or a shortage of nutrients can disrupt the delicate web of reactions that sustain life. And yet, even when disturbed, the biochemical systems of living beings strive to restore equilibrium. This property, the ability to maintain internal balance despite external change, is called homeostasis. It is life's most fundamental promise: to persist through change, to seek harmony again and again. In the calm of night, when you breathe slowly and the world grows quiet, biochemistry continues its work. Cells exchange gases, muscles repair microscopic tears, the liver processes nutrients, and the brain consolidates memory. Sleep itself is a biochemical state, governed by molecules like melatonin and serotonin, each released in gentle sequence to guide the body toward rest. Even dreams are shaped by the movement of neurotransmitters, the chemical messengers that allow one neuron to whisper to another. In every phase of life, from waking to sleeping, the chemistry within carries on with serene consistency. Throughout history, scientists have sought to uncover these hidden workings. In the 19th century, the study of metabolism, the sum of all chemical reactions within living organisms, began to reveal that the processes of life could be explained in chemical terms. Yet even today, biochemistry remains a science of wonder. The more we understand it, the more profound the mystery becomes. For behind every molecule lies another level of complexity, another layer of structure, another question, waiting patiently for discovery. Perhaps what makes biochemistry so soothing to contemplate is its continuity. Life began through chemistry, and it endures through chemistry still. Every living cell carries within it echoes of the earliest molecules that once floated in Earth's ancient seas. The hydrogen in your body is as old as the stars. The oxygen in your lungs was once part of oceans, glaciers, and storms. In this way, biochemistry connects us not only to the living world but to the entire cosmos. It reminds us that the line between life and matter is not a boundary but a bridge. As we begin this gentle study, let us hold that thought quietly, that life, in all its complexity, is made from the simplest things: elements that bond and react, atoms that share electrons, molecules that organize themselves into patterns of astonishing elegance. Every heartbeat is chemistry in motion, every breath a cascade of molecular reactions. The purpose of biochemistry is not just to understand these processes, but to appreciate them, to see the harmony that lies within structure, the calm precision of life unfolding at every scale. And so, as we close this first chapter, let the idea settle softly in the mind that you are a constellation of molecules, a living pattern of reactions that has persisted for billions of years. The atoms within you have been part of oceans, mountains, and stars, yet now they move together in the rhythm we call life. Biochemistry is the study of that rhythm, quiet, constant, and endlessly beautiful. 2. The chemical foundations of life. At the very core of life's structure and activity lies the language of chemistry, a universal system of elements, bonds, and interactions that has been in motion since the dawn of the universe. When we speak of living organisms, we are speaking of arrangements of matter that have found ways to organize, reproduce, and adapt, all through the quiet work of chemistry. The human body, like all living things, is built from a remarkably small collection of elements. Yet from this modest palette arises everything: the shape of your bones, the color of your eyes, the rhythm of your heart, and the thoughts that drift through your mind. The 6 elements most essential to life are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Together, they make up more than 99% of the atoms within all living things. The remainder, trace elements like iron, zinc, copper, magnesium, and iodine, play delicate but vital supporting roles, often as catalysts or cofactors in biological reactions. Each of these elements possesses a distinct character, a unique ability to form bonds and share electrons, which determines how they interact with one another. Their behavior follows predictable rules, but the outcomes of their combinations are almost infinite, forming the basis for all organic molecules. Hydrogen, the simplest element, rests at the beginning of this story. It contains just 1 proton and 1 electron, the most basic of all atomic arrangements. Yet it is everywhere in living matter. Hydrogen bonds are the invisible threads that weave molecules together, stabilizing the structures of DNA and proteins. This small atom plays an outsized role in the architecture of life. Oxygen, by contrast, is a voracious element. It attracts electrons strongly, a property that gives it the power to pull energy from other molecules during respiration. Every breath you take introduces oxygen into your bloodstream, where it quietly fuels the oxidation of nutrients, releasing energy in a form the body can use. Nitrogen, the element of proteins and genetic material, is another cornerstone. It forms the backbone of amino acids, the building blocks of proteins, and is found in the bases that spell out the genetic code of DNA. Though it makes up nearly 80% of Earth's atmosphere, nitrogen gas is remarkably inert. Only certain bacteria and lightning possess the ability to convert it into reactive forms that plants and animals can use. In this way, Nitrogen's cycle through the biosphere becomes a gentle reminder of chemistry's patience, transformation that takes time, guided by living intermediaries. Phosphorus, found in every molecule of DNA and RNA, connects one nucleotide to the next through sturdy phosphate bonds. These same bonds appear again in adenosine triphosphate, or ATP, the universal energy currency of the cell. Whenever a cell performs work, from moving a muscle to transmitting a nerve impulse, it spends ATP, breaking one phosphate bond and releasing a pulse of energy. Thus, phosphorus acts as both a builder of information and a carrier of energy, bridging two of life's most essential needs, memory and motion. Sulfur's role is quieter, but equally profound. Present in certain amino acids, such as cysteine and methionine, it helps shape the three-dimensional structure of proteins. Sulfur atoms form disulfide bridges, strong chemical bonds that lock protein chains into their functional forms. Without these crosslinks, enzymes and hormones would collapse into useless tangles. The scent of rain, the taste of onions, and even the resilience of keratin in hair and nails all owe something to sulfur's distinct chemical nature. And then there is carbon, the true architect of life. What makes carbon extraordinary is its flexibility. Each atom can form 4 bonds, allowing it to connect in an endless variety of ways. Chains, rings, branching structures all arise from carbon's quiet versatility. It can link with itself, forming the long molecular backbones that support the structure of organic compounds. Carbon's ability to form stable, yet reactive bonds enables the complexity necessary for life's chemistry. If hydrogen and oxygen are the rhythm of living systems, carbon is the melody that holds everything together. These 6 elements unite in countless configurations to create the molecules of life. When bonded with hydrogen and oxygen, carbon forms carbohydrates, sugars, and starches that serve as the body's energy sources. Combined with nitrogen, it gives rise to amino acids, the seeds of proteins. When joined with phosphorus, it forms the framework of nucleic acids, the carriers of genetic information. Each new combination adds another layer of intricacy, a new step in the long dance of living chemistry. What binds these elements together are the fundamental forces of atomic interaction. At the smallest scale, everything that exists is a product of attraction and repulsion between charged particles. Electrons orbit atomic nuclei, forming regions of probability where bonds are born. When 2 atoms share electrons, a covalent bond is formed. These are the strong, lasting connections that define molecules such as water, glucose, and DNA. Sometimes, electrons are not shared but transferred from one atom to another, creating ions that attract each other through opposite charges. This is the ionic bond, weaker in water, but essential in forming salts and regulating nerve impulses. Among these forces, one of the most subtle, yet powerful, is the hydrogen bond. It is not a true bond in the chemical sense, but a delicate attraction between polar molecules. Water, for instance, owes nearly all its remarkable properties to hydrogen bonding. These faint interactions give water its high boiling point, its surface tension, and its ability to dissolve so many substances. Without hydrogen bonds, the molecules of life could not maintain their shapes. DNA would unravel, and proteins would lose their structure. In the chemistry of living systems, small forces have enormous consequences. All of these interactions take place within the medium of water. It is often said that water is the matrix of life, and indeed, it serves as the stage upon which all biochemical reactions unfold. Each molecule of water is polar. One end carries a slight negative charge, and the other, a slight positive one. This polarity allows water to form an intricate network of interactions, continually breaking and reforming bonds in a dynamic balance. Within the cell, this constant movement keeps molecules dissolved, mobile, and ready to react. When water interacts with other molecules, It sorts them based on their affinity for polarity. Substances that dissolve easily in water are called hydrophilic, water-loving. These include ions, sugars, and most proteins. Other substances, such as fats and oils, are hydrophobic, water-fearing. They cluster together, forming membranes and barriers that define the boundaries of cells. In this way, water's polarity gives rise to one of biology's most fundamental structures, the lipid bilayer, a delicate film that encloses every living cell, separating the inside from the outside while allowing selective exchange. The interplay of these chemical forces gives rise to the complexity of life. Each molecule, each bond, contributes to a larger network of reactions, all working in quiet harmony. Yet none of it could exist without the universal laws of chemistry, the same laws that shape stars, oceans, and stones. The difference lies in organization. In living systems, matter does not simply react. It reacts with purpose, guided by enzymes, regulated by feedback, and sustained by energy flow. Energy itself is the thread that ties all biochemical activity together. The breaking and forming of chemical bonds is how energy moves through living systems. When bonds form, energy is released. When they break, energy is absorbed. The balance of these two processes defines metabolism, the sum of all chemical activity in a cell. This constant exchange ensures that life remains dynamic, never static. The energy once stored in sunlight passes through food, through molecules like glucose, and finally into ATP, a small, elegant molecule that serves as the universal currency of energy transfer. Though invisible to the eye, these molecular interactions are the true foundation of existence. They are the reason that matter can organize itself into cells, that cells can organize into tissues, and that organisms can sense, move, and think. When viewed closely, life is a tapestry woven from the simplest materials, electrons, protons, and neutrons arranged in intricate patterns of balance. Chemistry gives life its tools, but biochemistry gives it its meaning, transforming raw matter into awareness. As you rest and listen, imagine the universe at the level of atoms, each one a sphere of potential, surrounded by a cloud of motion. They do not stand still but vibrate, collide, and bond in an endless exchange of energy. From these movements arise the structures of your body, the rhythms of your heart, and the thoughts in your mind. The same atoms that once belonged to ancient stars now form the molecules of your being. The chemistry of life is not separate from the chemistry of the cosmos. It is its continuation, written in the same timeless language. And so, in the gentle quiet of this study, we find that life's foundation is both simple and profound. The elements that build us are ancient, the bonds that shape us are universal, and the energy that moves through us is eternal. To understand biochemistry is to see yourself as part of a great, unbroken chain of matter and motion, a harmony of atoms that has lasted for billions of years and will continue long after we are gone. The chemistry of life, steady and serene, is the silent story of the universe learning to know itself. 3. Water, the solvent of life. Of all the substances on Earth, water is perhaps the most unassuming, and yet it is the one most essential to life. It covers more than 2/3 of our planet's surface and fills the silent interiors of every cell that has ever lived. To speak of water in the context of biochemistry is to speak of life itself. Its presence defines the boundaries of what we call living, and its absence marks the limits beyond which no organism can survive. But water is not merely a backdrop for biology. It is an active participant in every reaction, every movement, every spark of vitality that flows through the body and the natural world. Imagine, for a moment, a single drop of water resting on a leaf in early morning light. To the eye, it appears still and simple. Yet within that droplet, Billions of molecules move with quiet precision, forming and breaking connections countless times each second. Each water molecule is composed of two hydrogen atoms bound to one oxygen atom, a simple formula, H2O, that belies its remarkable complexity. The oxygen atom, slightly more electronegative, draws the shared electrons of the bond closer to itself, creating a subtle imbalance of charge. The result is a polar molecule, one end faintly negative and the other faintly positive, like a tiny magnet adrift in space. This polarity is what allows water to perform its quiet miracles. Because of it, water molecules are attracted to one another, forming delicate hydrogen bonds that flicker in and out of existence with immense speed. Though individually weak, these bonds collectively give water its extraordinary properties, its cohesion, its surface tension, its ability to moderate temperature, and most importantly, its talent for dissolving almost anything it touches. This is why water is often called the universal solvent. It welcomes ions, minerals, and organic molecules into its embrace, separating and surrounding them, making it possible for chemical reactions to take place. Inside the human body, this dissolving power becomes the very foundation of life's chemistry. The fluids that flow through your veins, the sap within a tree, the plasma that carries nutrients to cells, all are water-based solutions. In these solutions, ions like sodium, potassium, and chloride drift freely, balancing electrical charges and regulating nerve impulses. Sugars and amino acids dissolve and travel where they are needed, while proteins fold into their intricate shapes within the watery environment of the cytoplasm. Without water, these substances would remain locked in place, inert and lifeless. It is the fluidity of water that allows life to remain dynamic. But water's gift is not only in movement, it also lies in stability. Because of its hydrogen bonds, water resists sudden changes in temperature. It absorbs heat slowly and releases it gradually, creating a thermal buffer that protects living organisms from extremes. Oceans and lakes remain temperate even as the air grows hot or cold, allowing marine life to thrive. Within the human body, this property ensures that internal temperature remains steady, preventing the fragile machinery of enzymes from faltering. Every warm-blooded creature owes its constancy to water's patient capacity to hold and share heat. The structure of water also makes it uniquely suited for supporting the architecture of life. When water freezes, its molecules arrange themselves into an open lattice, expanding rather than contracting. This makes ice less dense than liquid water, allowing it to float. It is a small and subtle difference, but one of profound consequence. If ice sank, lakes and oceans would freeze from the bottom up, trapping all aquatic life beneath. Instead, ice forms a protective layer at the surface, insulating the water below and preserving life through the coldest months. It is no exaggeration to say that entire ecosystems survive because of the unusual behavior of frozen water. In living cells, water shapes structure as much as it supports function. The proteins that perform nearly all of life's chemical tasks fold themselves in response to the watery environment around them. Portions of the molecule that are hydrophilic, water-loving, extend outward, interacting easily with the surrounding fluid, while hydrophobic, water-fearing regions curl inward, seeking shelter from contact. This interplay creates the three-dimensional shapes that give proteins their purpose. A similar process occurs in cell membranes, where phospholipids align themselves so that their hydrophobic tails face inward, protected from water, while their hydrophilic heads face outward. This simple arrangement, born from water's dual nature, becomes the defining structure of all living cells. Water also participates directly in countless biochemical reactions. Some reactions use it to split larger molecules apart, a process known as hydrolysis. Others produce water as a byproduct, such as when amino acids join to form proteins. In both cases, Water acts as a mediator, sometimes a tool of separation, sometimes a token of completion. Even the flow of energy within cells depends on water's presence. In photosynthesis, water molecules are split by sunlight, releasing oxygen into the atmosphere and electrons into the cycle that powers the plant's metabolism. In cellular respiration, the reverse occurs. Oxygen combines with hydrogen, forming water once again, as energy is released for use. Thus, through endless cycles of synthesis and breakdown, water flows at the heart of all biological energy. On the surface of the earth, water's influence is equally profound. It sculpts landscapes, nourishes forests, and carries minerals from mountain to ocean. It evaporates into clouds, condenses into rain, and returns again, sustaining the rhythm of the biosphere. The same molecule that forms the dew on a blade of grass may one day drift in a storm cloud or flow through the veins of a living creature. It moves endlessly, binding the living and non-living world together in a single continuous cycle. Every sip you take has traveled through rivers, roots, and bodies, linking you to every form of life that has ever existed. For biochemists, the fascination with water lies in how it makes possible both order and spontaneity. Within the liquid environment of the cell, molecules are constantly colliding, reacting, and rearranging. Yet out of this seeming chaos emerges balance. Reactions proceed only when conditions are right, and equilibrium is restored when they are not. Water is the medium that allows this to happen. It tempers the violence of chemical reactions, absorbs their excess heat, and provides the flexibility for life to adapt. In a sense, it is the peacekeeper of chemistry, maintaining calm amid constant motion. Water's behavior can even influence the structure of entire ecosystems. Plants, for instance, rely on the cohesion and adhesion of water molecules to transport nutrients from root to leaf. As water evaporates from the surface of leaves, it pulls more upward through tiny channels in the stem, a process called transpiration. This continuous column of water defies gravity, sustained by nothing more than the attraction between molecules and their affinity for the plant's inner walls. It is a delicate and silent force, yet it moves forests. The same principle governs blood flow, nutrient transport, and every form of circulation known to biology. In the realm of the microscopic, water's presence is even more intimate. The interior of a cell is not a chaotic soup, but a finely tuned solution, where concentrations of ions and molecules are carefully maintained. Here, water enables enzymes to recognize their substrates and catalyze reactions with astonishing precision. It forms thin layers around macromolecules, influencing how they fold, bind, and move. This structured water, neither entirely liquid nor entirely solid, acts as a bridge between the rigid world of molecular structure and the fluid realm of biological motion. In this middle ground, chemistry and life meet. What is perhaps most beautiful about water's role is its constancy across all scales. The same molecule that glistens in a raindrop performs the same duties within the nucleus of a cell. It bonds, separates, transports, and renews. Indifferent to size or setting, its properties are universal, its behavior timeless. In the great chain of existence, water is both the thread and the loom, weaving together molecules, cells, organisms, and ecosystems into one living tapestry. When viewed through the lens of biochemistry, water reveals itself not just as a physical substance, but as a principle, a symbol of balance and continuity. It connects the past to the present, the simple to the complex, the inert to the living. Every biochemical reaction, every pulse of energy, every act of renewal depends upon it. To understand water is to glimpse the unity of all life, bound by invisible forces and shared origins. As you rest your thoughts upon this image, picture the flow of water through you from the air you breathe to the blood that moves quietly beneath your skin. Each molecule carries within it a story older than life itself, shaped by stars and oceans, by evaporation and rain. You are, in a profound and literal sense, an extension of this eternal cycle. Biochemistry merely translates that truth into the language of atoms and bonds. Water, in its gentle persistence, is the silent witness to it all, the patient medium through which the miracle of life unfolds, moment by moment, breath by breath, forever in motion and forever at peace. 4. The building blocks. Biomolecules, all living things, from the simplest single-celled bacterium to the most complex human being, are built from a small family of organic compounds known as biomolecules. These molecules are the architects of life's form and the engineers of its function. Texture of muscle and skin carry the messages that guide growth, store the energy that fuels movement, and record the genetic instructions that make each individual unique. Though their names may sound technical, carbohydrates, lipids, proteins, and nucleic acids together, they create the quiet symphony of chemistry that sustains existence. To study them is to uncover the foundation upon which all life rests. Each biomolecule tells a different part of the same story, how matter organizes itself to become living. Yet what binds them all together is carbon, the same carbon that drifts through the atmosphere as invisible gas, that rests in the soil beneath your feet, that flows through oceans and roots and blood. Through carbon's remarkable ability to bond in endless combinations, life finds its flexibility. The diversity of living things is mirrored in the diversity of the molecules that compose them. Within this shared framework, the 4 major classes of biomolecules perform their intricate roles, each indispensable to the balance of the cell. Carbohydrates are often the first to come to mind when we think of biological energy. They are the sugars and starches that power muscles, feed the brain, and fuel every flicker of movement. At their core, they are composed of carbon, hydrogen, and oxygen, usually in a ratio that mirrors that of water. Simple sugars, known as monosaccharides, include glucose and fructose, small and sweet molecules that serve as immediate energy sources. When linked together, they form more complex structures, disaccharides like sucrose, the sugar found in fruit, or polysaccharides like starch and cellulose. In plants, starch acts as a reserve of energy, stored for times of scarcity. Cellulose, by contrast, provides structure, forming the sturdy walls that give plants their rigidity. In animals, glycogen performs a similar role to starch, accumulating in liver and muscle tissue to be released when energy runs low. Yet, carbohydrates are not merely fuel. They are also part of the cell's identity, adorning the surfaces of proteins and lipids like molecular signatures. These sugar chains, known as glycoproteins and glycolipids, act as identifiers, allowing cells to recognize one another and communicate. Through these gentle exchanges, tissues organize, immune cells distinguish friend from foe, and entire organisms maintain harmony among their countless parts. In every sense, carbohydrates are both energy and language, the nourishment of life and its means of connection. Lipids, on the other hand, tell a quieter story, one of structure and protection. Unlike carbohydrates, they do not dissolve in water. Instead, they gather together in soft films and membranes, creating the boundaries that define every living cell. The simplest lipids are fats and oils, formed from glycerol molecules joined to long chains of fatty acids. When these chains are saturated, filled with hydrogen atoms, they form solid fats, like butter or animal fat. When unsaturated, with one or more double bonds between carbon atoms, they remain liquid, like olive oil. These differences, invisible at the molecular level, influence the texture and behavior of cell membranes and stored energy reserves alike. In living organisms, lipids serve several vital functions. They provide long-term energy storage, yielding more than twice as much energy per gram as carbohydrates when metabolized. They act as insulation, preserving warmth in animals and cushioning delicate organs from impact. Most importantly, they form the structure of cell membranes, the fragile barriers that separate life from its environment. These membranes are made of phospholipids, molecules that resemble tiny tadpoles with a hydrophilic head that loves water and a hydrophobic tail that shuns it. When immersed in water, they arrange themselves naturally into bilayers, with the tails facing inward and the heads outward, creating a stable, self-healing surface. Within this thin film, countless reactions take place, channels open and close, and the delicate dance of life unfolds. Lipids also include steroids, a class of molecules with a distinctive ring structure. Among these is cholesterol, often misunderstood yet essential. Cholesterol gives membranes their flexibility and serves as the precursor for vital hormones such as estrogen, testosterone, and cortisol. Without it, cells would lose their resilience, and communication between organs would falter. In plants, a related group of compounds called phytosterols perform similar roles, reminding us once again of the shared chemical language that connects all living forms. If carbohydrates are life's quick energy and lipids its protective framework, then proteins are its true workers, the builders, messengers, and catalysts of every biological process. Composed of smaller units called amino acids, proteins fold into intricate shapes that determine their function. There are 20 different amino acids found in the human body, each with a unique side group that gives it distinct properties. Some are hydrophilic, others hydrophobic. Some carry a charge, while others remain neutral. When linked together in long chains, these amino acids form polypeptides, which then twist and fold into the final, functional protein. The range of protein functions is astonishing. Structural proteins such as collagen and keratin give strength to tissues, forming tendons, skin, and hair. Enzymes act as catalysts, accelerating reactions that would otherwise take centuries to occur. Hemoglobin carries oxygen through the blood, while antibodies defend against disease. Hormones like insulin regulate metabolism, ensuring that energy flows where it is needed most. Even motion itself, from the beating of the heart to the contraction of a muscle fiber, depends on proteins that slide and pull at the molecular level. What makes proteins particularly remarkable is their adaptability. Their shapes are not rigid but responsive, changing subtly as they interact with other molecules. This flexibility allows enzymes to recognize their specific substrates with exquisite precision, catalyzing only the reactions they are designed for. The relationship between an enzyme and its substrate is often compared to a lock and key, though a more accurate metaphor might be a gentle handshake, a meeting of forms that fit perfectly for just a moment before parting again. Through this endless sequence of encounters, proteins guide the chemistry of life with calm efficiency. Finally, there are the nucleic acids, DNA and RNA, the carriers of life's instructions. Within their delicate strands lies the information that determines every characteristic of an organism. DNA, or deoxyribonucleic acid, stores the genetic code in a sequence of 4 bases: adenine, thymine, cytosine, and guanine. These bases pair in a predictable pattern: A with T, C with G, forming the elegant double helix that has become the emblem of modern biology. RNA, or ribonucleic acid, serves as the messenger, carrying the genetic information from DNA to the cellular machinery that builds proteins. Together, they form the memory and voice of life, ensuring continuity from one generation to the next. The discovery of nucleic acids revealed that all living beings share a common molecular ancestry. The DNA within a leaf, a bird, or a human follows the same basic structure and uses the same code. What differs is only the sequence the particular order of bases that spells out each organism's unique instructions. From these simple letters arises the diversity of the living world, from coral reefs to forests to the human mind. In every case, it is chemistry that writes the story, molecule by molecule, base by base. Although these four classes of biomolecules, carbohydrates, lipids, proteins, and nucleic acids differ in structure and function, they are interdependent. Energy from carbohydrates is stored in lipids, transported by proteins, and regulated through information encoded in nucleic acids. In the cell, they form an elegant network, each part supporting the other. Even the smallest molecule plays its role with precision, and together they create the harmony that sustains life. It is humbling to consider that everything alive is composed of these same ingredients. The oak tree in a forest, the fish gliding beneath the water, and the human heartbeat are all built upon variations of the same molecular foundation. This unity reminds us that life's complexity is born not from the number of its components, but from the subtlety of their arrangement. Through the endless combinations of these building blocks, nature has composed every living form that has ever existed. As you imagine these molecules, the sugars, the lipids, the proteins, and the nucleic acids, picture them not as abstract shapes, but as dancers in a silent, continuous performance. Each movement, each connection, each separation adds meaning to the whole. Within every cell, this dance never stops. Even as you rest, the molecules within you continue their timeless work, building, repairing, communicating, and remembering. Biochemistry allows us to glimpse that hidden choreography and to understand, at least in part, how matter comes alive. The building blocks of life are not distant or foreign. They are the essence of your own being. Every thought, every heartbeat, every breath arises from their patient interactions. To study them is to recognize that within your body flows the same chemistry that shaped the stars, the oceans, and the first forms of life. The molecules that make you are ancient, yet their arrangement is uniquely yours, a quiet masterpiece of molecular architecture, sustained by the gentle balance of the biomolecules that make life possible. 5. Carbohydrates and energy flow. Among all the biomolecules that make up living systems, carbohydrates are the most immediate source of energy. They are the quiet engines of vitality that power every cell, from the smallest bacterium floating in a drop of water to the neurons that weave thought within the human brain. When we speak of carbohydrates, we speak of the molecules that connect sunlight to life, transforming the raw energy of the sun into a form that can be stored, transported, and shared among living things. Every breath, every heartbeat, and every movement of muscle fibers relies upon the steady, graceful flow of energy that carbohydrates make possible. Carbohydrates are composed of three elements: carbon, hydrogen, and oxygen, arranged most often in the ratio of one carbon atom to two hydrogens to one oxygen, a pattern that mirrors the formula of water. It is from this structure that their name arises: carbo for carbon and hydrate for water. Though simple in composition, they form a vast array of structures, from the single sugar molecules that dissolve sweetly on the tongue to the long, branching chains that give plants their strength. At their heart, all carbohydrates follow the same quiet logic: to capture and carry energy through the living world. The simplest of these molecules are the monosaccharides, or simple sugars. Glucose, fructose, and galactose are familiar examples, each containing the same number of atoms arranged in slightly different configurations. These tiny variations give each sugar its own subtle properties: how sweet it tastes, how it interacts with enzymes, and how it contributes to metabolism. Glucose, in particular, is the primary fuel of nearly every cell on Earth. It is the molecule that cells most readily recognize, the preferred source of quick energy. Once inside a cell, glucose is broken down through a series of gentle, stepwise reactions that release energy gradually in a form the cell can safely use. This process, known as cellular respiration, transforms the stored energy of food into adenosine triphosphate, or ATP, the molecule that powers nearly every biological task. The story of glucose begins long before it reaches an animal's bloodstream. It starts with plants, in the stillness of green leaves reaching toward the light. Through the process of photosynthesis, plants capture photons of sunlight and use that energy to combine water and carbon dioxide into glucose. This reaction not only builds food but also releases oxygen into the air, creating the conditions for all aerobic life to exist. The glucose molecules produced by plants become the foundation of the food web, feeding herbivores, carnivores, and omnivores alike. Thus, carbohydrates are the universal bridge between the energy of the sun and the vitality of life. When two monosaccharides join together, they form disaccharides. Sucrose, the common table sugar, is made of glucose and fructose, linked by a bond that holds potential energy, ready to be released when broken down during digestion. Lactose, the sugar found in milk, combines glucose and galactose, providing nourishment to newborn mammals in the earliest moments of life. These disaccharides remind us that even the simplest sweetness carries the weight of evolution, the biochemical memory of how living beings have learned to store and share energy. More complex carbohydrates, called polysaccharides, are formed when hundreds or even thousands of sugar molecules connect in long chains. The arrangement of these chains determines their role. Starch, found in plants, is composed of glucose molecules linked in a way that allows easy access for enzymes. It serves as a convenient energy reserve, ready to be broken down when sunlight fades or seasons change. Animals, too, store glucose, but in the form of glycogen, a more compact, highly branched molecule that can be mobilized quickly when energy is needed. Glycogen accumulates in the liver and muscles, quietly waiting for the signal to release its stored fuel. When you run, think, or breathe deeply, it is glycogen that yields glucose to sustain you. Some carbohydrates, however, serve not as fuel but as structure. Cellulose, perhaps the most abundant organic compound on Earth, forms the rigid walls that give plants their shape and strength. Though it is made entirely of glucose, the bonds between its molecules are arranged differently, creating long, unbranched fibers that resist digestion. Humans and most animals cannot break down cellulose, though it passes through our bodies as dietary fiber, aiding in the rhythm of digestion. But for herbivores like cows and termites, whose gut bacteria can unlock its energy, cellulose is both shelter and sustenance. In forests, it forms the wood that holds trees aloft. In the smallest grasses, it provides resilience and texture. Beyond structure and energy, carbohydrates play subtle roles in communication and recognition. On the surface of cells, chains of sugars are attached to proteins and lipids, forming unique molecular signatures. These glycoproteins and glycolipids act like identification tags, allowing cells to distinguish one another's self from non-self, friend from invader. The immune system, for instance, reads these sugar codes to identify foreign pathogens, while during development, cells use them to organize into tissues and organs. In this way, carbohydrates extend their function beyond nutrition and structure, entering the realm of information, quietly guiding the organization of life. When carbohydrates enter the body, the process of digestion begins to unfold with calm precision. Enzymes in the mouth, stomach, and small intestine work in sequence to break complex starches into simpler sugars. Each enzyme recognizes its target by shape, cleaving the bonds that hold glucose units together. The resulting sugars are absorbed into the bloodstream and carried to the cells that need them most. The brain, for example, consumes glucose almost exclusively as its source of energy, burning it continuously to maintain awareness, thought, and memory. Muscles, too, depend on glucose, especially during physical activity when quick energy is required. What is not immediately used is converted to glycogen or, in times of surplus, to fat for long-term storage. The metabolism of carbohydrates is a model of biochemical balance. When glucose levels rise after a meal, the pancreas releases insulin, signaling cells to absorb and store the excess. When levels fall between meals, another hormone, glucagon, instructs the liver to release stored glucose back into the blood. This interplay maintains homeostasis, ensuring that the body's energy supply remains steady and reliable. Disruptions to this balance, such as in diabetes, remind us of the delicate coordination underlying even the simplest act of eating. The regulation of sugar within the body is a constant, quiet negotiation between molecules, enzymes, and hormones, a conversation that continues without pause, from birth until death. Carbohydrates also play an integral role in metabolism beyond direct energy supply. They provide the carbon skeletons for synthesizing amino acids and fatty acids, linking all major biochemical pathways together. In plants, the carbon fixed through photosynthesis not only builds sugars but also forms the basis for the entire biosphere. Every fruit, leaf, and seed is a product of carbon chains that once began as invisible molecules of carbon dioxide. Through this transformation, carbohydrates embody the cycle of life from light to matter, from air to nourishment. On a planetary scale, carbohydrates represent nature's way of storing sunlight. Forests, crops, and algae act as vast batteries, converting solar energy into chemical bonds that can last for months or years. When animals feed upon these plants, the stored energy flows onward, nourishing entire ecosystems. Even after death, carbohydrates continue their work, breaking down in soil to enrich it for new growth. The energy that once fueled a single leaf may, in time, support countless lives. When viewed through the tranquil lens of biochemistry, carbohydrates reveal the simplicity and elegance of life's energy economy. They are produced by light, consumed by life, and returned to the earth, forming a cycle as ancient as the planet itself. They do not act with force or haste, but with constancy, a steady current beneath the surface of all living systems. As you consider the quiet work of these molecules, imagine the unseen threads of connection they weave. The bread on a table, the sap in a tree, the honey made by bees, and the glucose flowing through your veins. All are variations of the same elemental pattern. Through them, energy moves gently from sun to soil, from plant to animal, from cell to cell, and finally, to the breath that leaves your lungs. The flow never ceases. It merely changes form. In every living being, carbohydrates carry the memory of light. They are the physical embodiment of energy captured from the stars, transformed into the sweetness of fruit, the strength of wood, and the motion of life. Each molecule is a small act of continuity, a bridge between energy and matter, between the past and the present. In their endless cycles of formation and breakdown, we find the quiet truth that sustains all biology, that life is not separate from energy, but its expression. And within every cell, that expression continues, patient and eternal, as the soft chemistry of carbohydrates keeps the rhythm of life alive. 6. Lipids, the silent protectors. If carbohydrates are the quick and lively carriers of energy, then lipids are the quiet, enduring guardians of life's structure and stability. They move without haste, performing their duties in silence, storing warmth, shaping boundaries, and shielding the fragile machinery of cells from the harshness of the outside world. Lipids do not dissolve in water as sugars do. Instead, they drift apart, forming layers, droplets, and membranes that define the limits of every living being. Within this stillness lies their power. They are the molecules of protection, subtle, strong, and deeply essential. At their core, lipids are a diverse family of hydrophobic molecules, meaning they repel water. This single characteristic gives rise to a remarkable range of biological roles. They are the oils that coat feathers, keeping birds dry in the rain. They are the waxes that seal leaves against evaporation. They are the fats that cushion organs, insulate against cold, and serve as reservoirs of energy for times when food is scarce. Even the boundary between the inside and outside of every cell, the cell membrane is built from lipids, forming a barrier so thin it can scarcely be imagined, yet so strong that it sustains life itself. Most lipids are composed of the same simple elements as carbohydrates: carbon, hydrogen, and oxygen, but their structure differs in a way that changes everything. Where carbohydrates mix easily with water, lipids resist it, and this resistance allows them to form compartments and barriers. The simplest lipids are fats and oils, also known as triglycerides. Each is made of one molecule of glycerol attached to 3 long chains of fatty acids. These fatty acids are like threads of carbon and hydrogen, sometimes straight, sometimes bent, depending on the nature of the bonds that link the carbon atoms together. When all the bonds between carbon atoms are single, the chain is said to be saturated, meaning it holds as many hydrogen atoms as possible. Saturated fats are solid at room temperature, forming the dense, creamy texture of butter or animal fat. When one or more bonds are double, the chain bends slightly, preventing the molecules from packing closely together. These are the unsaturated fats, liquid at room temperature, found in plant oils such as olive or sunflower oil. This difference, simple in chemistry, is profound in biology, shaping everything from the consistency of membranes to the flow of energy within the body. In living organisms, fats serve primarily as energy stores. Each gram of fat contains more than twice the energy of a gram of carbohydrate or protein, making lipids the most efficient form of fuel. Animals store fat in specialized cells called adipocytes, which cluster together in soft, pale tissues beneath the skin and around vital organs. This stored fat provides not only energy but also insulation, helping to maintain body temperature in cold environments. In hibernating mammals and migratory birds, these reserves are life-saving, sustaining them through long periods without food. Even in humans, the gentle layer of fat beneath the skin serves as both protector and preserver, shielding the body and regulating warmth. But lipids are not merely storage molecules. Their most extraordinary role lies in the formation of membranes. Every living cell, from the simplest bacterium to the most complex animal, is enclosed by a delicate sheet made primarily of phospholipids. Each phospholipid is a molecule with 2 personalities. One end, the head, loves water and is called hydrophilic. The other end, the tail, fears it and is hydrophobic. When placed in water, phospholipids spontaneously arrange themselves so that their heads face outward, touching the water, while their tails turn inward, away from it. The result is a bilayer 2 parallel sheets of molecules that seal themselves into a closed surface. This simple, self-assembling structure is the foundation of life's architecture. It forms the walls of cells and the compartments within them, keeping the internal environment separate from the external world. Within this thin film, only a few nanometers thick, lies the power of organization, the ability to maintain homeostasis, to regulate what enters and leaves, to create order from the surrounding chaos. The phospholipid membrane, ions, nutrients, and signals pass in measured exchange, ensuring that each cell remains balanced and alive. Without this quiet boundary, the complexity of living systems would dissolve into disorder. Interwoven within these membranes are other lipids that lend them stability and flexibility. Cholesterol is one such molecule. Though often misunderstood, cholesterol is vital to life. It slips between the phospholipid molecules, preventing them from packing too tightly in the cold or drifting too far apart in the heat. In doing so, it keeps membranes fluid yet firm, resilient yet responsive. From cholesterol, the body also crafts steroid hormones, molecules like estrogen, testosterone, and cortisol, which carry messages that regulate growth, reproduction, and metabolism. A single cholesterol molecule can be transformed into compounds that influence nearly every organ and process within the body, showing how lipids, though simple in structure, can hold immense biological power. Another remarkable class of lipids is the waxes, long, sturdy molecules that form protective coatings on plants, insects, and animals alike. The shine on a leaf, the waterproof sheen on a bird's feathers, the subtle gloss of human skin, all are gifts of waxes. They act as natural barriers, keeping moisture in and the elements out. In deserts, wax coatings prevent water loss from plants that endure the scorching sun. In cold oceans, whales and seals rely on lipid layers beneath their skin, known as blubber, to maintain body heat. These adaptations reveal the quiet versatility of lipids. They insulate, preserve, and protect without fanfare. Lipids also serve as messengers within the intricate communication networks of the body. Some are converted into signaling molecules that guide inflammation, healing, and cellular growth. Others, such as prostaglandins, regulate smooth muscle contraction and blood flow, coordinating responses that keep the body in balance. Even the brain relies on specialized lipids to form the myelin sheath, the insulating layer around neurons that allows electrical impulses to travel swiftly and efficiently. Without these lipid-based coatings, the signals of thought and motion would falter, slowing the elegant rhythms of perception and action that define life. The beauty of lipids lies in their subtle duality. They are both rigid and fluid, both protective and dynamic. In the quiet chemistry of the cell, they allow for separation and communication at once. While the lipid membrane encloses and shields, it also serves as a stage for proteins that act as channels and receptors, letting the outside world touch the interior of the cell in carefully regulated ways. This delicate balance between closure and openness, between structure and movement, is what allows life to exist as both stable and adaptable. Beyond their roles in the body, lipids are central to the larger cycles of the biosphere. In plants, oils accumulate in seeds, providing nourishment for germination and for the animals that eat them. In marine ecosystems, microscopic algae produce vast quantities of lipids that sustain entire food webs. These molecules, rich with energy, flow through ocean currents, feeding plankton, fish, and whales alike. Even after death, lipids persist, slowly breaking down into the organic materials that enrich soil and sediment. They are, in a sense, nature's quiet archives, carriers of sunlight, stored and passed forward through generations. From a biochemical perspective, the persistence of lipids is both a blessing and a reminder of time's slow patience. Their hydrophobic nature makes them resistant to decay, allowing them to endure where other molecules dissolve. In the geological record, remnants of ancient lipids have been found in rocks billions of years old, faint chemical fossils that whisper of the earliest forms of life. They stand as evidence that, long before complex, planted organisms walked the earth, lipid membranes were already forming the boundaries of primitive cells, protecting the fragile spark of life from the surrounding world. To think of lipids is to think of quiet endurance. They do not rush like sugars or spark like enzymes, but they hold the form of life together with unbroken steadiness. Every heartbeat, every thought, every breath occurs within the protection of lipid walls. The warmth that spreads through the body on a winter night, the gleam of a leaf in the rain, the soft curve of a droplet of oil, all are expressions of lipid chemistry. They are the still guardians of biology, the unseen architecture of vitality. As we reflect on their role, we begin to sense that lipids embody one of nature's most profound truths, that strength does not always come from speed or force, but from quiet persistence. The cell membrane does not fight to protect the cell. It simply exists, forming a barrier that endures. Fat does not clamor for recognition. It simply waits, patient and ready to give energy when the time comes. Lipids teach us that protection and preservation are acts of stillness, that even the calmest structures can hold the greatest power. And so, in the grand symphony of biochemistry, while other molecules move and react, lipids remain the steady rhythm beneath it all, the silent protectors that make the song of life possible. 7. Proteins, nature's artisans. Proteins are the quiet artisans of life, the versatile craftsmen that shape, build, and maintain every living structure. They are as essential to the body as the stars are to the night sky, countless, varied, and beautifully intricate. If carbohydrates are the energy carriers and lipids the protectors, then proteins are the doers, the molecules of action. They construct, they catalyze, they signal, and they defend. Within the stillness of a single cell, thousands of proteins work in unison, each performing a specific task in a choreography so precise that it borders on the miraculous. To understand proteins is to glimpse the quiet mechanics of existence itself, where form and function merge into one. Every protein begins as a simple chain of smaller units called amino acids. There are 20 types of amino acids commonly used by living organisms, and each one contains the same basic structure: a central carbon atom, an amino group, a carboxyl group, and a variable side chain. It is this side chain, unique to each amino acid, that determines its character. Some are drawn to water, others avoid it. Some carry an electrical charge, others remain neutral. When these amino acids link together through peptide bonds, they form a polypeptide chain, a long string that at first appears unremarkable. Yet from this simplicity arises a complexity beyond measure. The sequence of amino acids, encoded by the genetic instructions in DNA, dictates how the chain will fold into its final three-dimensional form, and that shape determines what the protein will do. This folding process is one of nature's most astonishing feats. It happens spontaneously, guided only by the chemical properties of the amino acids themselves and the gentle push of the surrounding environment. Some sections of the chain coil into spirals, known as alpha helices. Others form pleated sheets that stack together in layers. Gradually, the chain twists and folds, seeking stability in the precise arrangement of its parts. When complete, the protein may resemble a cluster, a ribbon, or a complex machine with moving parts. Its surface is covered in grooves, pockets, and contours that fit other molecules with remarkable accuracy. This exactness of shape is what allows proteins to recognize their partners, to bind, to react, to communicate. Each one is a tiny sculpture, crafted by chemistry, yet purposeful, as if by design. In the quiet economy of the cell, proteins serve as both workers and architects. Structural proteins give shape and strength to tissues. Collagen, the most abundant protein in animals, forms the scaffolding of skin, tendons, and bone, lending them resilience and elasticity. Keratin builds hair, nails, feathers, and horns, creating protective layers that resist wear. Elastin stretches and recoils, allowing arteries and lungs to expand and contract without damage. Each of these proteins is built to endure, their long fibers woven into the very fabric of life's architecture. They do not move or react quickly. Instead, they persist, silent and steady, maintaining the form that other molecules depend upon. Other proteins act as enzymes, the catalysts that make life's chemistry possible. Without them, even the simplest reactions would proceed too slowly to sustain life. Enzymes work by lowering the activation energy required for a reaction to occur, allowing it to unfold smoothly and swiftly. Each enzyme is tailored to a specific task, often binding to just one type of molecule called a substrate. The fit between an enzyme and its substrate is so precise that it has been likened to a lock and key. Yet this metaphor, though simple, understates the elegance of the interaction. When an enzyme and its substrate meet, the enzyme often changes shape slightly to accommodate its partner, embracing it like two hands, meeting in perfect alignment. Within that gentle contact, bonds are broken, new ones are formed, and molecules are transformed. This subtle chemistry governs nearly every process in the body. In the digestive system, enzymes break down food into nutrients. In cells, they manage the production of energy, the synthesis of DNA, and the removal of waste. Each step in these pathways is guided by a specific enzyme, and the chain of reactions continues without pause, maintaining the delicate balance of metabolism. Enzymes never tire and are not consumed in the process. When one reaction is finished, they are ready for the next, repeating their work endlessly. They are the quiet laborers of a life, unseen, yet indispensable. Proteins are also the messengers that carry signals across the vast distances of the body. Hormones such as insulin and growth hormone are proteins that circulate through the bloodstream, telling cells when to absorb nutrients or when to divide and repair. On the surface of cells, receptor proteins act as gatekeepers, recognizing these messages and responding accordingly. When insulin binds to its receptor, it triggers a cascade of events inside the cell, guiding glucose from the blood into storage or use. This ability to sense and respond is what allows the body to maintain equilibrium, a constant conversation among proteins, carried out in silence and precision. Transport proteins add another dimension to this versatility. Hemoglobin, perhaps the most famous of them all, binds oxygen in the lungs and releases it where it is needed, carrying the breath of life to every cell. Each hemoglobin molecule contains iron, which gives blood its red color and its ability to capture oxygen with ease. In the same way, other proteins transport nutrients, ions, and hormones across membranes and throughout the bloodstream. Even within cells, motor proteins walk along molecular tracks, carrying vesicles and organelles to their destinations. They move not with gears or motors, but with the flexing of protein limbs, powered by the breakdown of ATP. At this microscopic scale, motion is quiet but constant, a steady pulse of molecular work that keeps the machinery of life running. Proteins also defend. Antibodies, crafted by the immune system, are specialized proteins that recognize and neutralize foreign invaders, such as bacteria and viruses. Each antibody is exquisitely specific, recognizing a unique molecular pattern on its target. Once bound, it marks the intruder for destruction or blocks its harmful effects. This adaptability gives the immune system its remarkable precision, allowing it to remember and respond to threats across a lifetime. The structure of antibodies is a testament to the creative potential of protein chemistry, a flexible framework capable of generating millions of variations from a single genetic blueprint. Beyond their biological duties, proteins embody the principle of cooperation. Rarely does one act alone. Enzymes form complexes, structural proteins weave into networks, and receptors interact with signaling pathways to achieve coordinated outcomes. Inside a cell, this cooperation resembles an orchestra. Each protein plays its part, contributing to the greater harmony of metabolism, structure, and regulation. The rhythm of these interactions sustains life not through noise or force, but through balance, a continual interplay between precision and fluidity. The beauty of proteins lies in their impermanence as much as in their creation. They are constantly being built, used, and broken down, recycled into new forms as the needs of the cell change. In the liver, old enzymes are dismantled to make room for new ones. In muscles, fibers are repaired and strengthened in response to motion and rest. This cycle of renewal reflects life's ongoing effort to maintain order amidst change. Every molecule of protein has its moment, performing its duty before giving way to the next, each act contributing quietly to the persistence of the whole. It is humbling to consider that the sensations of life, movement, thought, emotion, depend on these silent artisans. When you open your eyes to the morning light, it is proteins in your retina that capture the photons. When your heart beats, proteins slide against one another to create contraction. When you remember a sound, it is proteins in your neurons that shape and strengthen the connections between cells. Every aspect of being alive is, at its core, the work of proteins, each one shaped from the same 20 amino acids, arranged in endlessly unique patterns. As we look deeper into the world of biochemistry, proteins reveal themselves not only as molecules of structure and function, but also as symbols of life's creativity. From the simplicity of their ingredients arises an infinite diversity of forms and purposes. They teach us that complexity need not come from chaos, but from the careful arrangement of simple parts working together in harmony. In this sense, proteins are more than matter. They are a language through which life expresses its intent, a timeless conversation between atoms and energy. In the stillness of the body, where countless reactions unfold each second, proteins remain the ever-present artisans, shaping the living world from within. They build, repair, and adapt, weaving together the countless threads of existence into a single, coherent whole. To understand them is to see that the quiet strength of life comes not from grandeur but from precision, from the endless patience of molecules that never rest. Proteins are the art within biology, delicate, purposeful, and eternal in their rhythm, the true craftsmen of nature's design. 8. Enzymes, the catalysts of life. In the quiet inner world of every cell, reactions are constantly unfolding, countless molecular exchanges that sustain the living state. Nutrients are broken down, energy is released, molecules are built, and waste is removed. Yet, on their own, these chemical reactions would proceed too slowly to support life. They would linger in stillness, awaiting chance collisions of molecules and the rare alignment of energy required to make bonds break and form. What transforms this chemical potential into motion is the presence of enzymes, the remarkable catalysts that make life's pace possible. Without enzymes, the body would be like an instrument forever waiting to be played. With them, the rhythm of life flows effortlessly, continuous and serene. Enzymes are proteins by nature. Each one is folded into a precise three-dimensional shape, forming a landscape of grooves, ridges, and valleys where molecules can meet. The place where this meeting occurs is called the active site, a small pocket carved perfectly to fit the enzyme's specific partner, known as the substrate. The fit between them is not rigid, but flexible and responsive. When the substrate enters, the enzyme subtly shifts its shape, embracing the molecule as though molded around it. This gentle interaction lowers the energy barrier that would otherwise prevent the reaction from occurring. The result is transformation, a bond broken, a new bond formed, and the substrate changed into a product that drifts away, leaving the enzyme untouched and ready to act again. What makes enzymes extraordinary is not only their efficiency, but also their selectivity. Each enzyme recognizes only its intended substrate, ignoring the millions of other molecules that might surround it. This specificity arises from the geometry of the active site, where every atom is positioned to complement its target. It is as if the enzyme holds a single key to a single lock, opening just one pathway among infinite possibilities. And yet, even with this narrow focus, enzymes act with breathtaking speed. Some can catalyze thousands of reactions every second, turning the slow chemistry of matter into the fluid chemistry of life. Consider, for a moment, the simple act of breathing. Oxygen enters the lungs and travels through the bloodstream, where it fuels the oxidation of glucose in the cells. This process, known as cellular respiration, unfolds in dozens of steps, each one requiring an enzyme. Glycolysis, the first stage, breaks glucose into smaller fragments. The citric acid cycle continues the process, releasing electrons that carry energy to the electron transport chain. At each step, a different enzyme stands ready, guiding the transformation, ensuring that no energy is lost and no harmful byproducts accumulate. The end result is adenosine triphosphate, or ATP, the molecule that powers every heartbeat, every thought, every act of renewal. Without enzymes, none of this would occur within the timescale of life. Enzymes not only accelerate reactions, but also bring order to them. In the dense, liquid environment of the cell, countless molecules float and collide. Without control, their interactions would lead to chaos. Enzymes act as guides, channeling these random encounters into meaningful sequences. Each enzyme is part of a larger network, one reaction feeding the next in a graceful flow. When one pathway speeds up, another adjusts to match it. When one slows, others compensate. This self-regulating harmony maintains the balance of metabolism, ensuring that the cell remains in homeostasis, neither in excess nor in deficiency, but perfectly poised. At the heart of enzymatic action lies the subtle interplay between energy and structure. Every chemical reaction requires a certain amount of activation energy to begin the small push that allows bonds to rearrange. Enzymes lower this barrier, not by adding energy, but by stabilizing the transition state, the fleeting moment when old bonds are breaking and new ones are forming. In doing so, they make the improbable probable, the difficult easy. It is a quiet act of facilitation, like guiding a ball over a hill with a gentle hand instead of letting it struggle upward alone. The enzyme does not change the final outcome of the reaction, it simply makes it happen sooner, smoothly, and with less resistance. Though enzymes are astonishingly efficient, they are also delicate. Their activity depends on the conditions of their environment, temperature, pH, and the concentration of ions and molecules. Each enzyme has its own range of comfort, within which it performs perfectly. Too much heat can unravel its folded shape. Too little can slow its motion to stillness. The wrong pH can alter the charges that hold its structure together. This sensitivity reflects the precision of life's chemistry. Just as a symphony requires every instrument to be tuned, the cell must maintain its internal environment to keep its enzymes in harmony. The regulation of enzymes is one of the most intricate aspects of biochemistry. Some are always active, performing their tasks continuously. Others lie dormant until they are needed, switched on by specific signals. This control can occur through molecules that bind to the enzyme and alter its shape, activators that enhance its function or inhibitors that suppress it. Feedback mechanisms ensure that no reaction proceeds too far. When a pathway produces enough of its final product, that product may return to inhibit the enzyme that started the process, closing the loop. In this way, enzymes not only create balance but also sustain it, forming a self-regulating system as elegant as any design in nature. There are enzymes that work alone, and others that require helpers. These helpers, known as cofactors and coenzymes, are small molecules or ions that assist in the reaction. Some are metals, such as zinc, magnesium, or iron, which help stabilize the active site. Others, like vitamins, carry chemical groups or electrons from one enzyme to another, linking reactions together in seamless continuity. Without these partners, many enzymes would remain inactive, incomplete. Together, they form a partnership, the enzyme providing structure, the cofactor providing flexibility, a union that allows life's chemistry to expand far beyond what proteins alone could achieve. Enzymes are also masters of adaptation. In different environments and organisms, they evolve to meet specific needs. The enzymes that break down food in the acidic depths of the stomach are not the same as those that operate in the neutral conditions of blood or the alkaline interior of a plant cell. In deep-sea bacteria that live near volcanic vents, enzymes withstand temperatures that would destroy those of most animals. In Arctic fish, enzymes remain active in near-freezing waters. This adaptability reveals the resilience of life's chemistry, the ability to persist, to refine itself, and to find equilibrium under any circumstance. Perhaps one of the most remarkable aspects of enzymes is their subtlety. Their work is invisible, their motion microscopic, yet their impact encompasses all of life. Every emotion, every movement, every act of thought depends on their quiet labor. When you digest a meal, enzymes unlock the energy hidden within it. When you heal from a wound, enzymes knit tissues back together. When you breathe, when you dream, when you sleep, enzymes guide the chemistry that allows each of these states to exist. They are the invisible artisans of transformation, present in every cell, performing their tasks with tireless grace. Enzymes also embody the principle of efficiency without waste. They act precisely where and when they are needed, leaving no trace of their work except the change they bring about. Their elegance lies not in grandeur but in subtle repetition, a molecule meeting its substrate, catalyzing a reaction, releasing the product, and beginning again. Millions of times each second, this cycle repeats, steady and unbroken, like a pulse that keeps the rhythm of life. In the silence of the molecular world, enzymes are the heartbeat of biology. If one were to imagine the living cell as a city, enzymes would be its workers, invisible yet essential, each assigned to a specific task. Some build, some break down, some transport, and some regulate. Together they maintain the continuous exchange of energy and matter that defines life. And though they never rest, they act without noise or waste, following only the subtle geometry of their design. To contemplate enzymes is to sense the elegance of nature's smallest details. They remind us that the power of life lies not in force but in precision, not in speed but in coordination. Each enzyme is a testament to the harmony between structure and function. A tiny, perfectly folded piece of order amid the ceaseless motion of the universe. Through their action, chemistry becomes life, and the still matter of the world finds purpose. Enzymes are the unseen mediators between the possible and the actual, transforming potential into being, one reaction at a time. And in their endless, silent work, they sustain the gentle continuity of existence itself. 9. Nucleic acids, the molecules of memory. In every living cell, from the simplest bacterium drifting in a drop of water to the complex architecture of a human body, there lies a quiet library of instructions. This library is written not on paper or parchment, but within molecules so small that billions can fit within a single cell. These are the nucleic acids, DNA and RNA, the keepers of life's memory. They are the recorders of lineage, the carriers of inheritance, and the silent architects of form and function. Without them, there would be no growth, no repair, no evolution, and no continuity between one generation and the next. They are the molecules that remember. Deoxyribonucleic acid, known to the world as DNA, is perhaps the most remarkable molecule ever discovered. It holds the blueprint of life in the sequence of its components. A code, so simple yet so profound, that it defines the difference between a fern and a whale, between a human being and the smallest of microbes. DNA is composed of 4 fundamental building blocks, called nucleotides, each consisting of 3 parts: a phosphate group, a sugar molecule known as deoxyribose, and one of 4 nitrogen-containing bases: adenine, thymine, cytosine, and guanine. The bases are the letters of life's alphabet, and their sequence spells out the instructions that guide every biological process. The beauty of DNA lies not only in its composition but in its structure. 2 strands of nucleotides wind around each other like a pair of entwined ribbons, forming the iconic double helix. The bases on one strand pair precisely with those on the other. Adenine always joins with thymine, and cytosine always with guanine. These pairs are held together by delicate hydrogen bonds, allowing the strands to separate easily when the time comes for the information to be read or copied. This complementarity ensures that the genetic message can be faithfully duplicated, each strand serving as a template for the creation of its twin. Inside the cell, DNA resides in the nucleus, coiled tightly around proteins called histones to form chromatin. During cell division, this material condenses into chromosomes, the carriers of heredity. Each chromosome holds thousands of genes, and each gene corresponds to a specific instruction, a recipe for building a protein. These instructions are not active all at once. They are turned on or off according to the cell's needs. Some cells, like those in muscle or bone, use only a fraction of the total genetic library, while others may access different portions. Yet the entire code remains present in every cell, an unbroken thread of information stretching from the first cell of an organism to its last. The process by which DNA's information is used to create proteins is both simple in concept and exquisite in execution. When a cell needs a particular protein, the relevant section of DNA unwinds and exposes its code. A complementary molecule, ribonucleic acid, or RNA, is then synthesized from this template in a process called transcription. RNA is similar to DNA but differs in a few key ways. It contains the sugar ribose instead of deoxyribose. It is single-stranded rather than double, and it replaces the base thymine with uracil. Once the RNA copy, known as messenger RNA or mRNA, is made, it carries the information out of the nucleus and into the cytoplasm, where the cell's machinery translates it into a chain of amino acids, the building blocks of proteins. This flow of information from DNA to RNA to protein is often described as the central dogma of molecular biology. It is the universal process by which life expresses its code, turning stored information into action. The mRNA acts as a messenger, but it does not work alone. It joins with a ribosome, a complex structure made partly of ribosomal RNA, which reads the sequence of the message 3 bases at a time. Each triplet of bases, called a codon, corresponds to one of the 20 amino acids used in protein synthesis. Another form of RNA, called transfer RNA, brings the correct amino acids to the ribosome, matching each codon with its partner through complementary base pairing. Slowly, step by step, the ribosome links the amino acids together, forming a growing protein chain that folds into its final shape. Through this elegant choreography, the instructions written in DNA are brought to life. The enzymes that catalyze reactions, the structural proteins that form tissues, the hormones that regulate growth, all arise from the code contained in nucleic acids. This process unfolds with astonishing precision and efficiency. Every second, in every cell of the human body, thousands of new proteins are synthesized according to the same timeless principles. Each reaction is a quiet affirmation of continuity, the ancient language of nucleotides translated into the living motion of biochemistry. Nucleic acids are not only blueprints, they are also the agents of inheritance. When a cell divides, its DNA must be copied so that each daughter cell receives an identical set of instructions. This replication begins when the 2 strands of the double helix unwind, exposing their bases. Enzymes called DNA polymerases then move along each strand, matching new nucleotides to their partners and creating 2 identical double helices. The process is remarkably faithful, with proofreading mechanisms that correct errors as they occur. Yet small mistakes, mutations occasionally slip through. Some are harmless, some beneficial, and others harmful, but all contribute to the gradual evolution of life, the slow shaping of species over time. Through mutation and selection, nucleic acids not only preserve the past, but also enable the future. RNA, though often overshadowed by DNA, plays its own vital and versatile role in the symphony of life. In addition to carrying messages, RNA molecules can fold into complex shapes that allow them to catalyze reactions, much like enzymes. These catalytic RNAs, or ribozymes, suggest that in the distant past, before proteins evolved, life may have relied on RNA both to store information and to perform chemical work. This ancient RNA world, as scientists call it, may have been the dawn of biochemistry itself, a world where strands of RNA replicated and evolved, eventually giving rise to the DNA protein systems that define modern life. In viruses, nucleic acids take on yet another form of existence. Many viruses store their genetic material as RNA rather than DNA, using host cells to replicate themselves. Some, like retroviruses, reverse the usual flow of information by converting their RNA back into DNA, inserting it into the host's genome. These interactions blur the line between life and chemistry, reminding us that nucleic acids are not merely passive carriers of data, but dynamic participants in the evolutionary process. Through their adaptability, they bridge the gap between the living and the non-living, between what is and what might become. The scale of what nucleic acids accomplish is almost beyond comprehension. In each human cell, the DNA, if stretched end to end, would measure about 2 meters in length. Yet it is folded so intricately that it fits into a space invisible to the naked eye. Within that space lies approximately 3 billion base pairs of information, a code that, if printed, would fill thousands of volumes. And within that code lies the entire history of an individual, the inherited wisdom of countless generations distilled into a molecular script. Despite its complexity, DNA is not static. It responds to the environment, bending to the needs of the organism. Genes can be activated or silenced depending on diet, stress, temperature, and countless other factors. These changes, though not altering the underlying sequence, can affect how the information is read, a phenomenon known as epigenetic regulation. Through subtle chemical modifications, such as the addition of methyl groups to DNA or the alteration of histone proteins, cells fine-tune the expression of their genes. It is as though the book of life can be read in different tones, its meaning shifting with circumstance, yet its words remaining the same. At its heart, the story of nucleic acids is one of memory and continuity. They hold within them the record of the past, the mechanism of the present, and the potential for the future. Every strand of DNA in your body is connected, through an unbroken chain of replication, to the earliest forms of life on Earth. The atoms that compose it have cycled through oceans and stars, yet the structure they form has remained constant across billions of years. In this way, nucleic acids are more than molecules. They are the embodiment of time itself, carrying forward the song of existence in an unending refrain. To contemplate them is to see the unity of life, the same molecular language written in the genes of a tree, a fish, or a human being. The same bases, the same code, the same mechanism of expression. Every heartbeat, every breath, every thought is guided by patterns first etched into the earliest living matter, and though the letters of the code are simple, their arrangement gives rise to all the diversity and wonder that the world contains. In the stillness of night, as your body repairs and renews itself, these molecules of memory are at work, copying, translating, and preserving the instructions of life. They ask for no recognition, no applause, only the chance to continue. Through their quiet persistence, life endures, adapts, and evolves a continuous thread woven through time, linking the first spark of existence to the consciousness that now reflects upon it. Nucleic acids are not merely the chemistry of life, they are its memory, its continuity, and its song, written in the universal language of molecules that never forget. 10. The flow of energy in cells. Within every living cell, energy flows as softly and continuously as a stream through a quiet valley. It is unseen, intangible, yet it is the force that animates all life. Without energy, no cell could move, divide, or grow. The gentle pulse of energy keeps the heart beating, the neurons firing, and the leaves of plants stretching toward sunlight. Every act of life, from the smallest chemical reaction to the vast cycles of ecosystems, depends upon the transformation and transfer of energy. The story of biochemistry is, in many ways, the story of how life captures, converts, and uses energy in harmony with the laws of nature. Energy, at its essence, is the capacity to do work. In living systems, this work takes many forms, moving ions across membranes, building molecules from simpler ones, transmitting signals, or maintaining temperature and structure. But unlike machines that burn fuel in bursts, life operates with delicate balance. It neither wastes energy nor stores it in excess. Instead, it maintains a steady flow, adjusting moment by moment. This constancy is made possible through the quiet orchestration of countless chemical reactions, each one regulated and interconnected, forming a network of life's metabolism. The foundation of energy flow in biology begins with sunlight. Nearly all energy used by living organisms can be traced back to the radiance of the sun. Through photosynthesis, plants, algae, and certain bacteria capture this energy and convert it into chemical form. In the still green cells of leaves, light strikes molecules of chlorophyll, exciting their electrons to higher energy states. This energy is then used to drive reactions that transform carbon dioxide and water into glucose, a simple sugar that stores the captured sunlight in its chemical bonds. Oxygen, a byproduct of this process, is released into the air, enriching the atmosphere and enabling respiration in animals and humans alike. In this way, photosynthesis becomes the first great movement of energy from the inanimate world into the living one. Glucose, formed by plants and consumed by animals, becomes the central currency of cellular energy. Within each cell, glucose enters a series of reactions designed to extract its stored energy, gradually, efficiently, and safely. The first stage of this process is glycolysis, a sequence of 10 enzyme-catalyzed steps that take place in the cytoplasm. Here, a single molecule of glucose, containing 6 carbon atoms, is gently broken down into 2 smaller molecules of pyruvate. Along the way, a small amount of energy is captured in the form of ATP adenosine triphosphate, and high-energy electrons are transferred to carrier molecules like NADH. Even though glycolysis yields only a small portion of glucose's potential energy, it is enough to sustain simple organisms and to serve as the foundation for more complex processes in others. If oxygen is available, the pyruvate molecules formed in glycolysis move into the mitochondria, the energy centers of the cell. Within these tiny organelles, the next phase unfolds, the citric acid cycle, also known as the Krebs cycle. This elegant series of reactions completes the breakdown of glucose, releasing carbon dioxide as a byproduct. Each turn of the cycle generates additional carriers filled with high-energy electrons, stored in molecules such as NADH and FADH2. These carriers are the couriers of energy, transporting it to the final stage of the process, where the real transformation occurs. The final stage, called oxidative phosphorylation, takes place along the inner membrane of the mitochondrion. Here lies the electron transport chain, a series of protein complexes that act like stepping stones for electrons. As the electrons move from one complex to another, their energy is used to pump hydrogen ions across the membrane, creating a gradient, a difference in concentration, that stores potential energy like water behind a dam. When these ions flow back across the membrane through a protein called ATP synthase, their movement drives the formation of ATP from ADP and inorganic phosphate. It is a graceful process, quiet yet powerful, turning the energy of sunlight, captured long ago by plants, into the universal energy currency of life. ATP is perhaps the most important molecule in all of biochemistry. Often called the molecular battery, it carries small, manageable amounts of energy to where it is needed. Each ATP molecule stores energy in the bonds between its phosphate groups. When one of these bonds is broken, energy is released and used to power cellular functions. In the blink of an eye, ATP fuels muscle contractions, nerve impulses, and the synthesis of DNA and proteins. Yet ATP is not hoarded. It is created, used, and recreated continuously, a cycle that never stops. A single human cell may use and regenerate millions of ATP molecules every second, a testament to the ceaseless flow of energy within the body. Not all organisms rely on oxygen to generate ATP. Some, living in the depths of the ocean or the dark soils of the earth, perform anaerobic respiration, using alternative molecules like nitrate or sulfate to accept electrons. Others rely solely on glycolysis, producing energy through fermentation. Yeast, for instance, converts glucose into alcohol and carbon dioxide, while muscle cells under strain produce lactic acid. Though these processes yield far less energy than oxidative phosphorylation, they allow life to persist even in oxygen's absence. In this adaptability lies the resilience of biochemistry, the ability to find a way in every environment, no matter how harsh or limited. Energy, once produced, must also be managed carefully. Too much energy released at once could damage the delicate structures of cells, while too little would bring them to stillness. The balance is maintained through a series of feedback systems that regulate every step of metabolism. When energy is abundant, cells slow the breakdown of glucose, storing excess energy in the form of glycogen or fat. When energy is scarce, these stores are tapped, releasing fuel back into the bloodstream. Enzymes act as gatekeepers, responding to signals from hormones like insulin and glucagon, ensuring that every cell receives what it needs without waste or excess. This coordination extends throughout the body, linking every organ in the shared rhythm of energy use and renewal. Beyond the boundaries of the individual, energy flows through entire ecosystems in much the same way. Plants capture sunlight, herbivores consume plants, carnivores consume herbivores, and decomposers return nutrients to the soil, closing the cycle. In each step, energy is transformed from light to chemical, from chemical to motion, from motion back to heat never lost, only changed. The warmth of sunlight, the hum of a bee's wings, the pulse of a heartbeat, and the quiet decay of fallen leaves all belong to the same great current of energy that began when the first rays of the sun touched the surface of a young earth. Inside your own body, this current flows unbroken. The food you eat carries the memory of sunlight captured by distant plants. As it moves through digestion and metabolism, its energy becomes your own. The movement of your muscles, the thoughts in your mind, the warmth of your skin. Every breath you take draws in oxygen that once belonged to a tree, and every exhalation releases carbon dioxide that will, in time, return to another leaf. In this gentle exchange, the boundaries between self and world dissolve, and you become part of the same energy that moves through all living things. The elegance of this flow lies in its constancy. Energy is never destroyed. It only changes form, cycling endlessly through matter. Biochemistry, in its quiet precision, manages this flow, transforming raw energy into life and life back into energy again. It is a perfect balance of giving and receiving, of motion and rest. The mitochondria within your cells, descendants of ancient bacteria that once lived freely in the primordial oceans, continue this process as they have for billions of years. In every heartbeat and every breath, they echo the same rhythm, the rhythm of sunlight transformed into being. As we reflect on the flow of energy in cells, it becomes clear that life is not a static condition, but a constant act of transformation. To live is to move energy from one state to another, to channel the potential of the universe into motion, thought, and renewal. Every spark within you is part of a larger cycle that began long before your birth and will continue long after. Energy does not belong to any one form of life. It merely passes through, pausing for a time to illuminate each being before moving on. In that gentle passage lies the profound continuity of existence, a quiet, unending river of energy that sustains the miracle of life. 11. Cellular metabolism and pathways. Deep within every living cell, far beyond what the eye can see, lies a world of ceaseless motion. Molecules drift, collide, and react in an intricate dance of transformation. This invisible choreography is called metabolism, the sum of all chemical reactions that sustain life. It is through metabolism that food becomes energy, energy becomes motion, and motion gives rise to the continuous renewal of cells and tissues. In this quiet and perpetual activity, the cell keeps itself alive, adapting and adjusting in harmony with its surroundings. Metabolism is not chaotic, but beautifully organized, a vast network of pathways where each molecule follows a purpose and each reaction leads seamlessly to the next. At its heart, metabolism serves two intertwined purposes: to build and to break down. These twin aspects are known as anabolism and catabolism. Catabolic pathways release energy by breaking larger molecules into smaller ones, while anabolic pathways use that energy to construct new molecules necessary for growth and repair. Together, they form a balanced system, a living equilibrium, where creation and destruction occur simultaneously. Just as the tides rise and fall yet maintain the sea, so too does metabolism sustain life through cycles of use and renewal. Catabolism begins when complex molecules such as carbohydrates, fats, and proteins are consumed or released from storage. Each of these macronutrients enters a specific pathway that converts its chemical bonds into usable energy. Carbohydrates are broken down through glycolysis into smaller fragments called pyruvate, which are then further processed in the mitochondria. Fats are oxidized into fatty acids and glycerol, which in turn enter their own cycles of breakdown. Proteins, when needed, are dismantled into amino acids, their nitrogen removed, and their carbon skeletons repurposed for energy. Though the routes differ, the destination is the same: the generation of ATP, the molecule that carries the cell's energy supply. The breakdown of nutrients follows a graceful sequence of events. After glycolysis, the pyruvate molecules formed from glucose enter the mitochondria, where they are transformed into acetyl-CoA, a small but highly energetic compound. This marks the beginning of the citric acid cycle, also known as the Krebs cycle, which serves as the central hub of cellular metabolism. Here, acetyl-CoA combines with oxaloacetate to form citrate, and through a series of reactions, carbon atoms are released as carbon dioxide, while high-energy electrons are captured by carrier molecules like NADH and FADH2. These carriers then transport their electrons to the electron transport chain, where their energy is finally converted into ATP. The entire process is elegant and efficient, each molecule passing energy to the next, like a line of lanterns illuminating the dark interior of the cell. The production of ATP is not merely a mechanical outcome, but a finely tuned process of balance. The energy stored in its phosphate bonds must be released gently, in increments small enough for the cell to use without damage. Too much energy at once would generate heat and chaos. Too little would bring the cell to stillness. Enzymes act as mediators, guiding each reaction, ensuring that every transfer of electrons, every movement of ions, occurs in measured steps. In this way, metabolism embodies the principle of moderation, the quiet art of converting energy without waste, of maintaining life without excess. Anabolism, the other side of metabolism, builds the structures that define and sustain the cell. It uses the energy provided by catabolism to synthesize complex molecules from simpler ones. From amino acids, the cell assembles proteins. From nucleotides, it constructs DNA and RNA. From fatty acids and glycerol, it forms the lipid membranes that shape and protect it. These anabolic reactions do not occur randomly. They follow specific pathways that mirror the logic of construction, one molecule serving as the foundation for another. Each addition, guided by enzymes that recognize exactly where and how to connect the pieces. The result is a living architecture that maintains itself, constantly repairing, renewing, and adapting. The interplay between catabolism and anabolism is maintained through an intricate network of regulation. Signals from within and outside the cell determine which pathways are active and which are dormant. When nutrients are plentiful, anabolic processes dominate, building reserves and new structures. When energy is scarce, catabolism takes precedence, breaking down stored materials to provide fuel. Hormones such as insulin and glucagon act as messengers in this system, coordinating metabolism across entire organs. Insulin encourages the storage of glucose as glycogen and the synthesis of lipids and proteins, while glucagon triggers their release when energy is needed. Together, they keep the body's internal chemistry in balance, ensuring that no resource is wasted and no cell goes unfed. Among the most fascinating aspects of metabolism is its cyclical nature. Many pathways form closed loops that renew themselves continuously. The citric acid cycle, for instance, consumes acetyl-CoA but regenerates its starting compound with each turn. The urea cycle converts toxic ammonia from amino acid breakdown into harmless urea for excretion, while simultaneously recycling key intermediates. Even photosynthesis and respiration, the great energy cycles of the biosphere, mirror one another in elegant reciprocity. The oxygen released by plants becomes the breath of animals, and the carbon dioxide exhaled by animals becomes the nourishment of plants. Through these endless exchanges, life maintains its balance, a vast metabolic web connecting every organism on Earth. In the still interior of the cell, metabolism is not uniform but compartmentalized. Different reactions take place in specific regions, ensuring efficiency and control. The mitochondria handle energy production. The nucleus directs synthesis through genetic information, and the endoplasmic reticulum assembles proteins and lipids. Vesicles transport materials from one compartment to another, and membranes act as both barriers and bridges. This spatial organization allows the cell to perform thousands of reactions simultaneously, without confusion, each proceeding in its own time and place. It is a silent orchestra of chemical events, where every instrument knows its role, and the music of life plays without pause. Metabolism is also adaptable, capable of shifting its pathways to meet changing conditions. In fasting or famine, the body turns from carbohydrate metabolism to fat oxidation, preserving glucose for the brain and red blood cells. In times of abundance, excess nutrients are stored for future need. Cells in active tissues, such as muscles, adjust their metabolism moment by moment, responding to the immediate demands of movement and rest. This flexibility allows organisms to survive fluctuations in environment and availability, turning metabolism into a living dialogue between the inner and outer worlds. At its deepest level, metabolism reflects the unity of all living things. The same pathways that break down glucose in human cells also operate in the cells of yeast, plants, and bacteria. The same ATP that fuels a human heartbeat also powers the motion of a leaf unfurling or a bacterium swimming through water. This shared chemistry speaks of a common origin, a single spark of life that learned to transform energy into order and pass that knowledge on to every descendant. Each living creature, no matter how complex or small, is a variation of the same theme matter animated by the same rhythmic exchange of molecules. To imagine metabolism is to imagine a vast landscape of reactions, each one flowing into the next like rivers merging into the sea. The molecules move, combine, and separate in an endless pattern of renewal. There is no waste, only transformation. No true beginning or end, only continuation. Even in death, metabolism persists in another form, the decomposition that returns nutrients to the earth, feeding new life once again. The cycle remains unbroken, a whisper of energy and matter that has echoed since the dawn of the first cell. In every heartbeat, in every breath, this ancient chemistry continues within you. The food you eat, the air you breathe, the light that warms your skin, all are drawn into the same great network of reactions that sustains the living world. Metabolism is the quiet pulse of life, the invisible thread connecting the microscopic to the immense. It reminds us that existence itself is not a fixed state but a continuous act of transformation. Each cell, through its endless chemical symphony, participates in the same ancient rhythm, the metabolism of the universe, the serene and unending flow of life in motion. 12. Homeostasis at the molecular level, beneath the surface of all living systems, beyond the complexity of organs, tissues, and cells, there exists a deeper harmony, a balance so precise and persistent that life itself depends on it. This equilibrium is known as homeostasis. It is the quiet order that allows living things to remain steady amid constant change, to adapt without collapse, and to endure in an unpredictable world. At the molecular level, homeostasis is the silent conversation between thousands of reactions happening at once, each adjusting to the other, maintaining an internal environment that is both fluid and stable. It is the chemistry of calm, a balance that keeps chaos at bay. In its essence, homeostasis means stability through motion. No cell is ever truly still. Its molecules are always shifting, binding, unbinding, building, and breaking down. Yet, despite this continual motion, the cell maintains constancy, a temperature within narrow limits, a pH finely tuned, an ion concentration carefully measured. The reason this is possible lies in feedback, the invisible loops of cause and effect that regulate the cell's activities. When something changes, when a molecule rises or falls in abundance, the system senses the shift and responds, returning itself toward balance. This delicate responsiveness is what separates life from matter. It is the difference between a stone that passively endures and a cell that actively preserves its inner world. At the molecular level, homeostasis is built upon sensors, signals, and responses. Specialized molecules act as detectors, perceiving changes in their environment, a drop in energy, a surge in acidity, a scarcity of nutrients. These sensors send chemical messages that activate or inhibit other molecules, setting off chains of reactions that restore equilibrium. Enzymes, those master catalysts of life, play a central role here, adjusting their activity according to the needs of the moment. When the concentration of a product grows too high, it may bind to the enzyme that produces it, slowing the reaction, a mechanism known as feedback inhibition. When supplies run low, the inhibition is lifted, and production resumes. It is a self-correcting rhythm, subtle and continuous, requiring no conscious direction. The control of energy within the cell is one of the finest examples of molecular homeostasis. ATP, the molecule that carries energy, must be kept at a nearly constant level, despite fluctuations in demand. When energy consumption rises, as in a contracting muscle or a firing neuron, enzymes that produce ATP are activated, and pathways that consume it are slowed. When energy is abundant, the balance reverses, preventing waste. This coordination ensures that the cell never exhausts its supply and never produces more than it needs. In this balance lies the serenity of metabolism, a harmony maintained by chemistry alone. Equally delicate is the regulation of pH. Most enzymes function only within a narrow range of acidity, and even small deviations can disrupt the shape and charge of their active sites. Within the cytoplasm, buffering systems made of weak acids and bases absorb excess hydrogen or hydroxide ions, keeping the pH stable. The bicarbonate buffer in blood is one such system. When carbon dioxide accumulates and dissolves in water, forming carbonic acid, the buffer absorbs the excess hydrogen ions, preventing dangerous drops in pH. When the pH rises, the reaction reverses, releasing hydrogen ions to restore balance. This simple chemical equilibrium, repeated in every living being, keeps the difference between life and dysfunction within the width of a breath. Temperature, too, must be held in check. Proteins, the molecular machines of life, are sensitive to heat, too much, and they unfold, losing their structure and function. Too little, and their motion slows to stillness. Organisms have evolved elegant strategies to moderate temperature: sweating, shivering, panting, or adjusting blood flow. Yet at the cellular level, the protection begins with molecules called chaperones. These specialized proteins help other proteins maintain or regain their proper shape when conditions fluctuate. They act as quiet guardians, folding and refolding the cell's machinery, ensuring that life continues smoothly, even when the environment wavers. Ion balance, particularly of sodium, potassium, calcium, and chloride, is another cornerstone of homeostasis. These ions create the electrical gradients that power nerve impulses, muscle contractions, and nutrient transport. The cell membrane maintains this balance through molecular pumps and channels that open and close with precision. The sodium-potassium pump, for example, moves sodium ions out of the cell and potassium ions in, consuming ATP to maintain the necessary difference in charge. This imbalance is not a flaw, but a feature, a stored potential that allows the cell to respond quickly when needed. The moment a nerve fires, this potential is released in a wave of electrical activity, only to be restored again seconds later. It is an elegant cycle of disturbance and repair, of action followed by calm. Homeostasis also extends to the level of molecular synthesis. When an enzyme or protein is no longer needed, it is degraded and recycled, its components used to make new molecules. DNA repair systems constantly scan the genetic material for damage, fixing errors before they accumulate. Antioxidant molecules patrol the cell, neutralizing free radicals that might otherwise harm vital structures. These defenses work silently, unnoticed, yet without them, the ordered patterns of life would quickly unravel. Each molecule knows its place, its role in maintaining the quiet continuity of the whole. In multicellular organisms, homeostasis expands beyond individual cells into a symphony of coordinated control. Hormones carry chemical messages through the bloodstream, linking distant tissues into one unified system. The brain, through the hypothalamus, acts as a conductor, sensing changes and directing responses. If blood sugar rises, insulin signals cells to absorb it. If it falls, glucagon releases stored energy. When the body overheats, blood vessels widen and sweat glands activate. When it cools, muscles tremble to generate warmth. Each of these actions arises from molecular interactions, receptors binding to signals, enzymes activating or silencing. Yet together they sustain a stability so complete that most of the time we never notice it. Homeostasis does not mean stillness. It means dynamic equilibrium, a balance maintained through movement. Every heartbeat is a small disruption, followed by a restoration. Every breath changes the composition of the blood, and every exhale returns it to balance. The chemistry of life depends on these fluctuations, for without them, adaptation would be impossible. The strength of homeostasis lies in flexibility, in the capacity to bend without breaking, to adjust without losing integrity. In this way, the living cell resembles a flame ever-changing, yet constant in form, maintained by the steady exchange of matter and energy. When homeostasis falters, the effects ripple through the system. If temperature regulation fails, enzymes misfold. If ion balance collapses, nerves fall silent. If pH drifts too far, reactions cease. Yet even in distress, the body's molecular systems strive to restore order. Fever, for instance, is not a failure of homeostasis, but a temporary adjustment, a deliberate raising of temperature to fight infection. Similarly, inflammation, swelling, and clotting are all controlled disturbances, protective measures designed to restore equilibrium. Life, it seems, depends as much on the ability to recover balance as to maintain it. At a broader scale, the principle of homeostasis extends beyond organisms to the Earth itself. The atmosphere, oceans, and living systems interact to regulate the planet's temperature, composition, and cycles. Carbon dioxide is absorbed by plants and oceans, oxygen is released, and nutrients circulate through soil and water. This planetary homeostasis, sometimes called the Gaia principle, reflects the same patterns seen within cells: feedback, regulation, and adaptation. Life not only maintains its own balance, but contributes to the stability of the world that sustains it. At the molecular level, homeostasis is a quiet triumph of precision. It is the art of remaining whole in a world of change, the unspoken agreement between countless molecules to cooperate for the sake of continuity. Even in sleep, even in silence, these processes continue, ions flowing, enzymes adjusting, signals traveling unseen. It is a reminder that beneath our awareness, an ancient and perfect balance sustains us, every second, without pause. To reflect on homeostasis is to glimpse the serenity at the heart of life's motion. Every heartbeat, every breath, every cell holds within it a microcosm of order that has persisted for billions of years. Life endures not through resistance to change, but through harmony with it. The molecules of the body do not fight the world. They listen to it, respond to it, and find balance within it. Homeostasis is their language, a song of stability woven from constant movement, a whisper of chemistry that says, again and again, stay. And through that whisper, the living world continues, calm amid the endless motion of existence. 13. The chemistry of life's origins. Long before there were forests, oceans teeming with fish, or even the faint shimmer of a living cell, the earth was a restless sphere of transformation. Its surface boiled and cooled, its atmosphere thick with gases, its seas dark and new. Yet within that apparent chaos lay the seeds of harmony, the first stirrings of chemistry that would one day give rise to life. Biochemistry, the study of living molecules, is also a window into this ancient beginning, a way to look backward and imagine the moment when matter first learned to organize itself. The story of life's origins is not a tale of sudden creation, but of gradual assembly, a patient unfolding of possibilities over countless ages, guided by the quiet laws of chemistry. In the earliest days of Earth, around 4 billion years ago, conditions were vastly different from those of today. The atmosphere was devoid of oxygen, filled instead with methane, ammonia, carbon dioxide, and water vapor. Lightning flashed across the sky, volcanic gases vented from the crust, and ultraviolet light from the young sun poured down without the shield of an ozone layer. In this harsh yet fertile environment, energy was abundant, and energy, as we now understand, is the essence of transformation. Where there is energy and matter together, reactions can occur, bonds can form and break, and complexity can grow. It was in such an environment that the first organic molecules appeared. These were not alive, not yet, but they were the building blocks from which life would eventually be woven. Experiments in the 20th century, such as the famous Miller-Urey experiment, showed that simple gases exposed to lightning-like sparks could produce amino acids, the same molecules that form proteins in living organisms today. From such beginnings, it is believed that sugars, lipids, and nucleotides also emerged, each formed through countless reactions, powered by heat, light, or the minerals of the early Earth. The oceans became vast laboratories, filled with a growing mixture of compounds, a primordial soup where chemistry was exploring its own possibilities. Yet having the ingredients was only the beginning. Life required not just molecules, but organization patterns that could store information, replicate, and evolve. Somewhere in those early waters, certain molecules began to interact in self-reinforcing ways. Fatty acids, for instance, have a natural tendency to form membranes when placed in water. Their hydrophobic tails cluster together, and their hydrophilic heads face outward, forming tiny bubbles called vesicles. These primitive boundaries could trap other molecules inside, creating a microenvironment, an early version of the cell's protective membrane. Within such vesicles, reactions could occur more efficiently, shielded from the turbulence of the outside world. The next great step was the emergence of molecules capable of replication. Among the simplest candidates for this role is RNA, a close chemical cousin of DNA, but more versatile. RNA can both store genetic information and catalyze reactions, a property that makes it a likely participant in life's earliest stages. Scientists propose that before DNA and proteins existed, there may have been an RNA world, where strands of RNA copied themselves with the help of surrounding molecules. Those that replicated more efficiently survived and multiplied, leading, over vast timescales, to greater complexity. Though fragile and imperfect, these first self-replicating systems marked the beginning of biological evolution, the transformation of chemistry into life. As time passed, these primitive systems grew more intricate. Some vesicles may have trapped strands of RNA inside, creating stable combinations of structure and information. Others developed rudimentary metabolic cycles, using surrounding chemicals to generate energy. Minerals such as iron and sulfur could have served as catalysts, accelerating reactions much like enzymes do today. Over millions of years, the merging of these functions, membranes, replication, and energy use gave rise to the first true cells. They were simple, no more than microscopic bubbles containing a few active molecules, yet they carried the defining feature of life, the ability to sustain and reproduce themselves. From this humble beginning, the long journey of evolution unfolded. Some cells learned to capture sunlight, using pigments to convert energy into chemical form, the ancestors of modern photosynthetic organisms. Others relied on consuming the organic molecules around them, a strategy that led to the diversity of metabolic pathways seen today. The waste products of these early metabolisms gradually altered the planet's atmosphere. Most significant of all was the rise of oxygen, produced by primitive cyanobacteria. At first, oxygen was toxic to many forms of life, but for others, it opened new possibilities: more efficient energy production, greater complexity, and eventually, multicellular life. The chemical principles that guided these ancient transformations are the same that govern biochemistry today. Carbon, with its ability to form four stable bonds, remains the foundation of life's architecture. Water, with its polarity and solvent properties, continues to serve as the medium for all reactions. Enzymes, though infinitely more sophisticated than the mineral catalysts of the past, follow the same logic, lowering the energy needed for reactions to occur. And the flow of energy, from sunlight to chemical bonds to motion and heat, still follows the same eternal cycle. The continuity between the earliest molecules and the cells of today is unbroken, a lineage of chemistry stretching across 4 billion years. To imagine the origin of life is to imagine patience beyond measure. There was no single moment of creation, no spark that instantly transformed matter into being. Instead, it was a slow gathering of processes, each step building on the last. Molecules formed, assembled, interacted, and refined themselves through trial and repetition. The Earth's environment provided both challenge and opportunity cycles of heating and cooling, drying and flooding, which concentrated molecules and drove them to react. In this sense, life emerged not by chance alone, but through the natural tendency of the universe toward complexity and order when energy flows through matter. Even today, the echoes of those ancient reactions remain within us. The membranes of our cells still follow the same basic structure as those first vesicles. The RNA molecules that carry messages from our DNA retain their ancient function as both carriers and catalysts. The cycles that convert nutrients into energy, such as glycolysis and the citric acid cycle, mirror chemical pathways that existed long before the first cell took shape. We are, in every sense, living fossils of those early experiments in chemistry, refined by time but faithful to our origins. It is humbling to realize that the atoms that compose us were forged in stars long before the Earth existed. Hydrogen, the simplest element, dates back to the birth of the universe itself. Carbon, oxygen, nitrogen, and phosphorus were created in the hearts of ancient suns, scattered across the cosmos by supernovae, and gathered into the forming Earth. Through countless transformations, they became part of oceans, rocks, air, and eventually, living cells. When you breathe, the oxygen in your lungs connects you to the stars that birthed it. When you drink water, you touch the same molecules that once cooled the newborn planet. The chemistry that sustains you is the same that shaped the universe. The origin of life, then, is not just a scientific mystery, but a reflection of continuity, a reminder that life and matter are not separate, but expressions of the same reality. Biochemistry reveals that the line between living and non-living is not a sharp divide, but a gradual gradient of complexity, a spectrum where organization deepens until it begins to sustain itself. Somewhere along that gradient, long ago, chemistry crossed the threshold into life, and from that moment onward, evolution began its long, unfolding story. When you look at a single cell under a microscope, you are looking at the descendant of that first spark of organization. Each movement within it, each molecule folded and refolded, each reaction flowing quietly into the next, carries the memory of its ancient origins. The same forces that shaped the first living systems still act within you now. You are the continuation of a process billions of years in the making, a stream of chemistry that learned to remember, to adapt, and to wonder about its own beginnings. In that thought lies a profound peace. Life did not arise from chaos, but from harmony, from the slow alignment of molecules, energies, and time. The same balance that holds your cells together was once written in the quiet chemistry of a young planet, waiting for the right moment to awaken. And in every heartbeat, in every breath, that ancient chemistry continues, steady, silent, and eternal, the first whisper of life, still echoing within the molecules that keep you alive. 14. The living cell. A universe within. Every living being, no matter how vast or small, is built upon the same foundation, the cell. It is the quiet center of biological existence, the smallest unit of life that can grow, divide, and sustain itself. Yet within this tiny structure lies an entire universe of activity, a world so complex and dynamic that it humbles even the most intricate human designs. The cell is both architect and builder, both machine and consciousness in miniature, performing countless chemical reactions with precision and grace. To look closely at a single cell is to glimpse the harmony of biochemistry, brought to life a balance of structure, energy, and purpose, sustained through endless motion. A cell is not merely a droplet of living matter, but a carefully organized community of molecules, each performing its role in perfect synchrony. It is bounded by a membrane that defines its limits, yet it is never truly isolated. Through that delicate barrier, it exchanges materials and information with the world beyond. Nutrients flow in, waste flows out, and signals arrive that influence its behavior. The membrane is composed of a double layer of phospholipids, their hydrophilic heads facing outward and their hydrophobic tails inward, forming a stable yet fluid boundary. Embedded within it are proteins that act as gatekeepers, channels, and receptors, molecular sentinels that allow the cell to sense and respond to its surroundings. In this soft, flexible wall lies the secret of life's adaptability, protection without rigidity, separation without isolation. Inside the membrane, the cytoplasm stretches like an ocean of life. It is not empty but filled with a clear, gel-like substance that holds enzymes, ions, and countless dissolved molecules in motion. Within this cytoplasmic sea float the organelles, small, specialized compartments that perform distinct tasks. Each organelle is like an island with its own purpose, yet none act alone. Together, they maintain the equilibrium that keeps the cell alive. In prokaryotic cells, such as bacteria, these structures are simple and open, with reactions occurring freely in the cytoplasm. In eukaryotic cells, those of plants, animals, and fungi, the internal landscape is far more elaborate, divided into membrane-bound regions that allow for greater specialization and control. At the heart of the eukaryotic cell lies the nucleus, the keeper of the genetic code. Surrounded by a double membrane punctuated by pores, it protects the DNA while allowing messages to pass through in the form of RNA. Within this quiet chamber, the blueprint of the organism is stored: long strands of DNA, coiled into chromosomes, each containing thousands of genes. When the cell requires a new protein, a section of DNA unwinds, and its information is transcribed into messenger RNA. This RNA leaves the nucleus and travels to the ribosomes, where the code is translated into the language of amino acids. The process is silent yet constant, a steady rhythm of reading and building, of information transformed into form. The ribosomes, tiny specks scattered through the cytoplasm or attached to the endoplasmic reticulum, are the factories of the cell. They assemble proteins with astonishing precision, linking amino acids one by one according to the instructions carried by RNA. Each completed protein folds into its unique shape and takes on its destined role as an enzyme, a structural element, or a signaling molecule. The endoplasmic reticulum, a vast network of folded membranes, assists in this task. In its rough form, covered with ribosomes, it produces and packages proteins. In its smooth form, it synthesizes lipids and helps detoxify the cell. Together, these structures form a continuous highway of molecular activity, connecting the nucleus to the outer membrane, ensuring that every part of the cell receives what it needs. Nearby, the Golgi apparatus acts as a postmaster of the cell, modifying and sorting the products of the endoplasmic reticulum before sending them to their destinations. Vesicles bud off its edges, carrying proteins to the membrane or to lysosomes, where they will perform their tasks. Lysosomes themselves are the recyclers, small acidic compartments filled with enzymes that break down worn-out components into simpler molecules, which are then reused. Through this process of renewal, the cell prevents decay and maintains efficiency. Nothing is wasted, everything is transformed. The mitochondria are perhaps the most remarkable organelles of all. Often described as the powerhouses of the cell, they convert the chemical energy of nutrients into ATP, the currency of life. Their inner membranes are folded into structures called cristae, where the electron transport chain carries out the delicate work of energy conversion. Mitochondria have their own DNA, distinct from that of the nucleus, a relic of their ancient origin as free-living bacteria that entered into partnership with early cells. This symbiotic relationship, formed billions of years ago, endures in every living creature today. The oxygen you breathe, the food you consume, all find their purpose within these tiny organelles, where energy is released to sustain your body. In plant cells, an additional organelle plays a central role, the chloroplast. These green structures capture sunlight and transform it into energy through photosynthesis, converting light into glucose and releasing oxygen as a gift to the world. Like mitochondria, chloroplasts possess their own DNA, reflecting their evolutionary heritage as ancient symbionts. Within their layered membranes, pigments like chlorophyll absorb light, initiating reactions that feed not only the plant, but all organisms that depend on it. Through these cycles of light and energy, the cell connects the smallest act of chemistry to the vast rhythms of the earth itself. Throughout the cell, the cytoskeleton provides shape and movement. It is made of protein filaments that stretch and contract, guiding vesicles, supporting membranes, and allowing the cell to divide or change form. Microtubules serve as tracks for motor proteins, while microfilaments give strength and flexibility. Even in stillness, the cytoskeleton is in motion, assembling and disassembling as needed. It gives the cell both structure and grace, enabling it to move, to reach, and to grow. The coordination of all these parts depends on communication, a molecular dialogue conducted through chemical signals. Receptors on the membrane detect hormones, nutrients, or changes in the environment, and relay messages inward through signaling pathways. These cascades often involve enzymes that activate one another in sequence, amplifying a small signal into a large response. In multicellular organisms, such coordination extends across tissues and organs, ensuring that every cell contributes to the harmony of the whole. Even single-celled organisms exhibit remarkable responsiveness, sensing light, temperature, or chemical gradients, and adjusting their behavior accordingly. In this way, the cell demonstrates a kind of awareness, a sensitivity born not from thought, but from the logic of chemistry. Despite its apparent complexity, the cell operates with remarkable efficiency. Every process, from the synthesis of proteins to the transport of ions, is regulated to minimize waste and maintain balance. When energy runs low, the cell slows its activities. When resources are abundant, it grows and divides. During division, the entire structure replicates itself with near-perfect fidelity. DNA copied, organelles distributed, membranes reformed. It is as if the cell contains not only the blueprint for life, but also the tools to perpetuate it indefinitely. Each new cell carries the memory of the one before it, a continuation of the same living stream. To see the cell in this light is to see it not as a mere collection of parts, but as a microcosm of the universe, self-contained yet connected, dynamic yet ordered. Its processes echo the principles that govern the cosmos. Energy flowing through structure, balance maintained through motion, creation born from transformation. Within each of your trillions of cells, the same chemistry unfolds at every moment, linking you to all living things that have ever existed. The cell is a testament to the unity of life, whether in the leaf of a tree, the wing of a bird, or the human heart. Its architecture follows the same ancient design. Its molecules are universal, its reactions timeless. It does not shout or boast of its complexity. It simply endures, performing its work with quiet precision. Every sensation, every breath, every heartbeat begins at this level within the membranes and molecules of living cells that have carried forward the spark of life since its earliest dawn. When one pauses to consider the cell, it becomes clear that life's wonder is not confined to the vast or the grand, but thrives in the smallest of spaces. A single cell holds within it the essence of all living things, a self-sustaining world of balance, creation, and renewal. It is both ancient and new, fragile yet enduring. Within its microscopic boundaries, the drama of existence unfolds continuously, a timeless rhythm that connects all life to its beginning. The cell is not only the building block of life, but also its quiet miracle, a universe within, whispering the eternal truth that even in the smallest space, infinity can dwell. 15. The harmony of biological systems life, in all its vastness and diversity, is not a collection of isolated parts, but a network of connections. A vast harmony in which each component supports and responds to the others. From the molecular dances within cells to the great cycles of ecosystems, everything in biology is part of a continuum, a symphony of interactions that sustains the living world. The quiet cooperation among molecules, organs, and species is what allows life to endure and adapt. When we look closely at this harmony, we begin to see that existence itself is built upon balance, balance between energy and order, between change and stability, between individuality and unity. At the smallest scale, this harmony begins inside the cell. The organelles within it are not independent entities, but participants in a shared rhythm. The nucleus provides instructions, the ribosomes build, the mitochondria supply energy, and the membranes communicate with the outside world. Each function depends on the others, creating a cycle of support that keeps the cell alive. When a single process falters, others adjust to restore equilibrium. This kind of self-regulating cooperation, so common in biology, mirrors the principle of harmony in music. When one note wavers, the surrounding tones shift to preserve the melody. In the same way, the cell maintains its song, its molecular symphony never ceasing. When cells join together, they form tissues, and the harmony expands. Muscle cells align in parallel fibers, contracting as one to create motion. Nerve cells form intricate networks that carry messages faster than thought. Epithelial cells build barriers and linings, defining the boundaries of organs. Each tissue performs a role, yet none can exist alone. Blood, flowing through every organ, carries nutrients, gases, and signals that coordinate their work. The heart, the lungs, the liver, and the brain, each performs its distinct rhythm, yet together they form the orchestration of the body. A single heartbeat relies on the synchronized firing of millions of muscle cells, each guided by electrical impulses shaped by ions moving across membranes. When those impulses align, the rhythm of life continues. When they falter, the harmony becomes dissonant. This balance between systems is known as physiological regulation. It ensures that every organ's activity matches the needs of the body. The nervous and endocrine systems act as conductors, sensing internal and external changes and adjusting responses accordingly. A rise in temperature triggers sweat and dilation of blood vessels. A drop invokes shivering and constriction. A surge of glucose after a meal prompts the release of insulin, guiding the sugar into cells for storage or energy. When the body is at rest, the parasympathetic nerves quiet the heart and slow breathing. When danger arises, the sympathetic system accelerates them, flooding muscles with oxygen and energy. These responses are seamless, automatic, and continuous, the body's own symphony of survival. The blood itself, is a carrier of harmony, linking distant regions through its constant circulation. It delivers oxygen to tissues and collects carbon dioxide to be exhaled. It carries hormones from glands to their targets, immune cells to sites of injury, and nutrients to cells in need. Within this crimson flow, billions of microscopic elements work in unison. Red blood cells, shaped for flexibility, pass through narrow capillaries. White blood cells patrol for signs of harm. Platelets wait patiently for wounds to heal. Each of these components acts independently yet cooperatively, sustaining the body's internal equilibrium. It is as if the bloodstream hums, with an unspoken awareness, every molecule in motion, part of the same enduring purpose. Harmony also extends beyond the body to the larger systems of nature. Just as organs sustain an organism, so do organisms sustain their environments. Plants absorb carbon dioxide and release oxygen, while animals do the reverse, completing the cycle of respiration that unites all breathing life. Microorganisms in the soil break down dead matter, returning its nutrients to the earth, which in turn nourishes new growth. Rivers carry minerals to the sea, clouds lift them skyward again, and rain renews the land. Each process, from the smallest exchange of ions to the movement of continents, contributes to a balance that allows life to flourish. Even decay is part of the harmony, a gentle return, a recycling of form into essence, ensuring that nothing is lost, only transformed. In ecosystems, this balance manifests through relationships that are both competitive and cooperative. Predators and prey shape each other's evolution. Pollinators and plants sustain mutual existence. Forests and oceans regulate the planet's climate. Beneath these visible patterns lies the same biochemical logic that governs a single-cell feedback, adaptation, and interdependence. When one part of the system changes, others respond. If nutrients become scarce, populations shift. If the climate warms, organisms migrate or evolve. Though such changes may seem turbulent, they are part of a deeper continuity, the planet's way of seeking equilibrium, of maintaining its living harmony across the ages. At the molecular level, even DNA participates in this pattern of balance and adaptation. It stores the memory of generations, yet it is never static. Mutations occur, sometimes harmful, sometimes beneficial, providing the raw material for evolution. Through natural selection, the living world refines itself, always adjusting to the conditions of existence. The same mechanism that allows a cell to repair its DNA after damage also enables species to adapt to new environments. The harmony of life, therefore, is not a fixed melody, but an evolving one, always finding new expressions while preserving its underlying theme. Within the human body, the brain is the supreme expression of biological harmony. Billions of neurons communicate through trillions of connections, transmitting signals as fleeting electrical sparks. These signals form thoughts, emotions, and memories, patterns of activity that mirror the balance and rhythm of chemistry itself. When you experience calm, it is the result of neurotransmitters aligning, of ions and receptors reaching equilibrium. When you think or dream, proteins in your neurons shift, enzymes act, and molecules rearrange themselves in patterns of meaning. The mind, in this view, is not separate from biochemistry, but its highest song, a harmony so refined that it becomes awareness. Even consciousness follows the same laws that govern metabolism and molecular reaction. It is sustained by the flow of ions, powered by ATP, maintained by homeostasis. Yet it rises beyond its components, a property that cannot be seen in the individual molecules, but only in their collective rhythm. In this sense, the human experience, thought, memory, feeling, is an extension of the same order that holds a cell together. We are both participants in and observers of life's grand harmony, capable of sensing the unity that underlies all complexity. This harmony, however, is not perfect in the sense of immutability. It thrives through flexibility, through the capacity to adapt and restore balance when disturbed. Illness, injury, and environmental change are all tests of this resilience. When one system falters, others compensate, striving to maintain the whole. The heart beats faster to deliver oxygen when lungs are weak. The liver detoxifies the blood when the kidneys cannot. In ecosystems, too, when one species declines, another rises to fill its role. Harmony in nature is not stillness, but the art of returning, of finding the center again and again, no matter how far one strays. To contemplate the harmony of biological systems is to see life as an unbroken continuum, from molecule to planet. The same rules that organize a single cell also govern the biosphere. The flow of energy, the exchange of matter, the feedback that sustains balance, all repeat at different scales, forming patterns that echo across the fabric of existence. We are not separate from this rhythm. We are part of it. Every breath, every heartbeat, every thought is a note in the universal composition of life. When we step back and see life in this way, complexity gives way to simplicity. The same chemistry that builds a flower builds a human being. The same sunlight that feeds the forest fuels the fire of thought. Beneath every difference lies a shared design, an ancient structure of balance and transformation. Life's harmony is not imposed from above but emerges from within, from the patient cooperation of countless small acts of order. And so, when you breathe deeply, when you feel the rhythm of your pulse, you are hearing a faint echo of that greater harmony, the whisper of the molecules that sustain you, the resonance of systems that began billions of years ago. Life is not a collection of separate lives, but a single, endless symphony, played across time and matter. Each organism, each cell, and each heartbeat adds its own tone, and together they form the serene and eternal music of existence. 16. The rhythm of evolution and adaptation. Life, from its simplest beginnings to the infinite variety that now covers the earth, has always been guided by change. The rhythm of evolution is the soft but unstoppable current that flows through all living things, shaping their forms, their functions, and even their behavior. Every organism alive today carries within it the memory of this unending process, the molecular traces of adaptation, the whispers of ancient survival, and the record of countless generations that came before. Evolution is not a story of sudden leaps or dramatic transformations. It is a quiet unfolding, a steady rhythm of adjustment and renewal that has continued for billions of years, sculpting the delicate harmony of life. The idea of evolution may seem vast and abstract, but its foundation lies in simple chemistry. Every living being is built from molecules that replicate, and wherever replication occurs, variation follows. When DNA copies itself, occasional errors, mutations appear, small changes in the sequence of bases that make up the genetic code. Most are neutral, some harmful, and a few beneficial. Those rare beneficial changes can improve survival or reproduction, allowing an organism to leave more descendants. Over time, these small advantages accumulate, and populations slowly shift, adapting to their environments. This process, known as natural selection, operates not with intention, but through the quiet mathematics of life itself. At the molecular level, evolution is a dialogue between stability and change. DNA is designed to preserve its message faithfully, yet it also permits just enough imperfection to allow innovation. Enzymes that copy DNA are precise but not flawless, introducing mutations that become the raw material for adaptation. Other processes, such as recombination during reproduction, shuffle genetic information, creating new combinations of traits. Even mobile elements, tiny segments of DNA capable of moving within the genome, contribute to this dynamic, rearranging the code and generating diversity. Through these molecular mechanisms, life experiments continuously, exploring the vast landscape of possibility written into its own structure. The earliest forms of evolution likely began long before complex organisms appeared. In the ancient oceans, simple self-replicating molecules competed for resources and stability. Those that could copy themselves more reliably or harness energy more effectively became more abundant. This primitive form of natural selection paved the way for the first cells, where cooperation among molecules gave rise to metabolism, protection, and self-repair. Over millions of years, cells diversified, some forming colonies that shared labor and resources. From these cooperative beginnings came multicellular life, an immense leap in complexity, achieved not through sudden invention, but through the slow rhythm of adaptation and interdependence. As life evolved, the environment itself became both teacher and sculptor. Climate, geography, and the availability of resources influenced every evolutionary path. When Earth cooled and oxygen filled the atmosphere, some organisms adapted to the new air, while others retreated to the depths where oxygen was scarce. The rise and fall of oceans, the drift of continents, and the coming of ice ages all acted as gentle yet relentless pressures, guiding evolution's course. In every case, the organisms that survived were not necessarily the strongest or the fastest, but those most attuned to balance, those whose chemistry could adjust to the changing conditions without losing its essential harmony. The rhythm of evolution is evident not only in the emergence of new forms, but in the quiet constancy of those that have remained unchanged. Certain bacteria, for example, have persisted almost identical for billions of years, their simple structures perfectly suited to their environments. Others, like the horseshoe crab or the ginkgo tree, carry the designs of ancient worlds, their forms stable and enduring. These examples remind us that evolution is not a race toward complexity, but a search for equilibrium, the meeting point where form and function align with the world's rhythms. Adaptation, the living heart of evolution, occurs in countless subtle ways. Some changes are physical, the lengthening of a limb, the thickening of a coat, the shaping of a beak. Others are chemical or molecular, invisible to the eye but profound in effect. Enzymes evolve to function at new temperatures or in different pH ranges. Membranes adjust their composition to remain fluid in heat or cold. Even the way cells process energy can shift, allowing organisms to thrive in extreme conditions. Deep beneath the sea, bacteria survive near volcanic vents, using sulfur instead of sunlight for energy. In frozen tundra, certain fish produce antifreeze proteins to prevent their blood from solidifying. Each adaptation is a quiet triumph of biochemistry, a small change in molecular design that transforms survival into mastery. Over long periods, the accumulation of adaptations leads to the diversity we see today. Evolution, when viewed from afar, resembles a great branching tree, with each branch representing a lineage, each leaf a species. Some branches grow lush and full, others fade and fall. Yet the tree continues to expand, rooted in the same origin. DNA comparisons reveal that all living things, from the smallest microbe to the tallest tree, share a common ancestry. The genetic code itself, the sequence of bases that spells life's instructions, is universal. This unity of code across the living world speaks of a shared beginning, a single rhythm that has echoed through every organism since the dawn of time. Within this grand continuity, humans occupy a unique but connected place. Our species is a product of the same evolutionary processes that shape all life. The structure of our hemoglobin mirrors that of whales and mice. Our enzymes share ancestry with those of ancient microbes. Even our brains, the seat of thought and creativity, are built upon patterns that evolved over millions of years, circuits refined from those that once controlled movement and sensation in simpler creatures. The ability to reason, to imagine, and to reflect upon our own existence is not an exception to nature, but its continuation. Consciousness itself can be seen as an adaptation, an internal mirror that allows life to observe its own unfolding. Evolution also carries with it a profound patience. It does not hurry or force change, it waits, adjusting gently as generations pass. The rhythm is so slow that it is almost imperceptible in a single lifetime, yet over eons, it transforms continents, ecosystems, and species. The coral reefs that build themselves grain by grain, the trees that reach higher with each century, the bacteria that adapt to new antibiotics, all are expressions of this same patient rhythm. There is a lesson in that quiet persistence, that life's strength lies not in speed, but in its capacity to endure and to adapt with grace. At the heart of adaptation is a dialogue between the organism and its environment. Every living thing is shaped by its surroundings, and in turn, it shapes them back. Plants alter the composition of the air, animals distribute seeds and nutrients, microbes recycle matter into usable form. Over time, these interactions become ecosystems, balanced systems of give and take that sustain both the individuals and the whole. When an ecosystem changes, so too must its members. Some evolve to fill new roles, others vanish, and still others emerge. Evolution is therefore not a solitary process, but a collective one, a reflection of life's interconnectedness across all forms and scales. The same principles that govern the adaptation of species also guide the evolution of ideas, societies, and even knowledge. Just as genes carry information that adapts to survive, so do thoughts and discoveries evolve, tested by experience, refined by understanding, and passed to new generations. Science itself follows an evolutionary rhythm, hypotheses arising, being tested, and giving way to better explanations. Through this ongoing process, humanity participates consciously in the very mechanism that created it, continuing the endless dialogue between stability and change, between what is known and what is yet to be discovered. In the grand flow of time, evolution reminds us that nothing in life is fixed, yet everything is connected. The molecules that once formed in ancient seas now compose your body. The genes that shaped early creatures continue to whisper their instructions in your cells. Adaptation is not a distant process, but one happening within you at every moment. As your body responds to temperature, stress, and the environment around you, life's rhythm moves through you, as it has moved through every being that came before, carrying forward the same message, that to live is to change, and to change is to endure. When you close your eyes and imagine the slow pulse of evolution, the turning of ages, the rising and falling of species, it feels like the heartbeat of the planet itself. The mountains shift, the oceans move, and within them, life adjusts, always seeking harmony with the world. From the first cell to the human mind, this rhythm has never stopped. It is the pulse of adaptation, the quiet force that binds all things in a single continuum of becoming. And through it, the chemistry of life remains forever alive, breathing through time, reshaping the world in silence, one gentle transformation at a time. 17. The interconnected web of life. All life on Earth exists within a vast, delicate web, intricate that no single thread can be touched without trembling through the rest. This web connects every organism, every ecosystem, and every molecule in a continuous cycle of exchange and renewal. It stretches from the smallest bacterium dwelling deep within the soil to the great forests that breathe out oxygen into the sky, from the whales gliding through the ocean's depths to the microscopic plankton drifting unseen beneath them. Each part, though small in itself, contributes to the balance of the whole. The interconnectedness of life is not merely a poetic image. It is the fundamental truth of biology. Within this network, energy flows, matter circulates, and life perpetuates itself in a rhythm that has endured for billions of years. When we look closely, we find that every living thing exists, not in isolation, but in relationship. The air we breathe was once exhaled by plants. The water in our blood once flowed through rivers and clouds. Even the atoms within our bodies, the carbon, nitrogen, and oxygen that form our tissues, were forged long ago in the hearts of stars. Life is, at its core, a continuous act of sharing. The boundaries we perceive between species, between organisms, and even between the living and the non-living, are fluid, part of an endless cycle of exchange. When a leaf falls to the ground, its elements return to the soil, feeding microbes and roots, becoming new leaves in seasons to come. Death, in this sense, is not an end but a transformation, a return to the greater cycle that sustains all existence. The flow of energy through this web begins with the sun. Through photosynthesis, plants capture sunlight and transform it into chemical energy, weaving light into matter. This stored energy becomes the foundation of nearly every food chain on Earth. Herbivores feed on plants, carnivores feed on herbivores, and decomposers break down what remains, returning nutrients to the soil and atmosphere. At each step, energy is transferred, transformed, and diminished as heat. Yet it never disappears. Matter, too, moves in cycles: carbon, nitrogen, phosphorus, and water, endlessly exchanged between the living and the non-living world. The carbon you exhale today may, in time, become part of a blade of grass, a coral reef, or a cloud drifting above a distant sea. Even within a single organism, this interconnectedness persists. The human body, for example, is not solely human. It is a thriving community of microorganisms, trillions of bacteria, fungi, and viruses that live in harmony within us. Together, they outnumber our own cells and play essential roles in digestion, immunity, and even emotion. They help break down food, synthesize vitamins, and protect us from harmful invaders. In return, we provide them warmth, nutrients, and shelter. This mutual dependence is a microcosm of life's greater web, showing that cooperation, as much as competition, is a fundamental principle of survival. In ecosystems, cooperation often takes the form of symbiosis, intimate relationships between different species that benefit one or both. Lichens, for instance, are composed of algae and fungi, living together so seamlessly that they function as a single organism. Coral reefs depend on microscopic algae that live within coral tissues, providing them with food through photosynthesis, while gaining protection in return. The roots of most plants form associations with fungi, known as mycorrhizae, that help them absorb nutrients from the soil. Even the oxygen we breathe is the product of collaboration, born from the partnership between ancient bacteria that learned to harness sunlight and the cells that later evolved to house them. The story of life is not one of solitary struggle, but of shared endurance, a chorus of cooperation echoing through time. The web of life extends far beyond the visible world. Beneath forests and grasslands lies a hidden network of fungal filaments, known as the mycorrhizal network, sometimes called the Wood Wide Web. Through these underground threads, trees and plants exchange nutrients and chemical signals. A shaded sapling may receive sugars from an older tree. A damaged plant may release distress signals that alert its neighbors to prepare defenses. These exchanges reveal that even plants, rooted and silent, participate in a community of awareness, responding to one another's needs. The same is true in the oceans, where vast blooms of plankton form the foundation of marine ecosystems, feeding creatures from the smallest fish to the largest whales. Every drop of water, every grain of soil, teems with life that depends upon the rest. This balance, however, is fragile. When one part of the web is disturbed, the effects ripple outward. The loss of a single species can unsettle an entire ecosystem, as the relationships that sustained it begin to unravel. The extinction of a pollinating insect can affect the flowers it fertilizes, the fruits that feed animals, and the seeds that would have grown into forests. The decline of coral reefs disrupts the habitats of thousands of marine creatures, diminishing the biodiversity of entire oceans. These examples remind us that the strength of life lies not in dominance or isolation, but in connection, in the gentle interdependence that binds every being to every other. Humans, too, are threads in this web, though we often forget our place within it. Our bodies and minds are products of nature, our survival tied to the balance of ecosystems. The food we eat, the water we drink, and the air we breathe are all gifts of the living earth. Yet our activities, the clearing of forests, the burning of fossil fuels, the pollution of oceans, strain this balance. The climate warms, species vanish, and the cycles of water and carbon shift in ways that threaten the stability that has nurtured us for millennia. In our pursuit of progress, we have forgotten that to harm the web is to harm ourselves, for we are not apart from nature, but an expression of it. Yet even now, the same resilience that has guided life through past extinctions and upheavals remains. Nature heals when given the chance. Forests regrow, rivers cleanse themselves, and species adapt to new conditions. The Earth's capacity for renewal is profound, but it depends upon balance, upon the respect for cycles that have sustained life since its beginning. When we act in harmony with these rhythms, we become not conquerors of nature but participants in its ongoing creation. The interconnectedness of life is also a reflection of its intelligence, not the intelligence of thought or language, but of pattern and adaptation. Every cell senses and responds. Every organism adjusts and evolves. Together, they form a network of awareness that spans the planet, responding to temperature, light, water, and time. The flight of migrating birds, the blooming of flowers, the spawning of fish, all follow cycles shaped by the turning of the earth and the flow of seasons. To live is to be attuned to these rhythms, to feel the pulse of the world moving through one's own heartbeat. When we understand life in this way, the distinctions between self and other, human and nature, begin to fade. The same molecules that make up your body move through rivers, clouds, and trees. The same energy that powers your cells once traveled from the sun to a leaf, and from that leaf to your hands. Life is not something you possess. It is something you share, a continuous current that flows through all things. Every breath you take connects you to the forest that made it possible, every meal to the soil and sunlight that gave it life. To see the world as a web of connection is to recognize the profound unity beneath diversity. Every living being, from the humblest bacterium to the most complex organism, is part of one continuous story, the story of Earth itself. This understanding brings with it not only knowledge but peace, a quiet realization that we belong to something vast and enduring. In every cell, in every breath, we participate in the same ancient symphony, the music of life interwoven across time and space. And so, as you imagine the interconnected web of life, picture it as a great tapestry of energy and matter, endlessly forming and reforming. Each strand glows softly, touching another, passing light from one to the next. The glow never fades, for even as one life ends, another begins, continuing the eternal cycle. The web holds everything from the smallest cell to the largest mountain, bound together in the same living thread. In that quiet connection lies the truth of existence, that nothing lives alone, and that in every moment, the whole universe breathes together as one. The chemistry of thought and emotion. Within the quiet chambers of the human mind lies one of the most extraordinary expressions of biochemistry thought. It is invisible, weightless, and fleeting. Yet it arises from the movement of molecules, the firing of neurons, and the delicate exchange of ions and neurotransmitters. Every sensation, memory, and emotion is born from the same molecular foundation that governs all living cells. The mind, for all its complexity, is a biological phenomenon, a symphony of chemistry and electricity that has evolved over millions of years to give rise to awareness. When we think, feel, and dream, we are not escaping the physical world, but participating in its most intricate form of organization. At the heart of thought lies the neuron, a specialized cell that communicates through electrical impulses and chemical signals. Each neuron connects to thousands of others, forming a network so vast that the human brain contains more connections than there are stars in the Milky Way. These connections, known as synapses, are the crossroads of thought. When a neuron fires, it sends an electrical pulse down its axon to the synapse, where it releases neurotransmitters, small chemical messengers that drift across a tiny gap to reach the next cell. There, they bind to receptors, triggering new electrical impulses that continue the signal. In this way, information moves not as a single stream, but as a vast constellation of pulses and exchanges, forming patterns that give rise to perception, movement, and consciousness itself. The chemistry of these neurotransmitters is both simple and profound. Dopamine, serotonin, acetylcholine, glutamate, and many others each play unique roles in shaping thought and emotion. Dopamine carries the feeling of reward and motivation, encouraging us to seek and act. Serotonin contributes to mood, stability, and calm. Acetylcholine governs attention and memory, while glutamate serves as the primary excitatory signal of the brain, the spark that keeps communication alive. These molecules are not abstract symbols of emotion, they are the emotion, the physical manifestation of joy, focus, or sorrow. A single change in their concentration can shift the tone of a day, coloring perception in subtle yet profound ways. The balance among these chemicals is delicate, and the body maintains it with remarkable precision. When the brain senses excess, it reabsorbs neurotransmitters or breaks them down through enzymes. When it senses deficiency, it produces more. This constant regulation ensures that thought remains fluid, not chaotic, and that emotion flows without overwhelming the self. Just as the heart maintains a steady rhythm through the balance of ions, the mind maintains stability through the interplay of molecules. It is a dance of equilibrium, where chemistry becomes consciousness and balance becomes peace. Memory, too, arises from the chemistry of connection. When we experience something new, neurons form or strengthen synapses, creating physical traces of experience. These connections, known as neural pathways, allow the brain to recall information long after the original event has passed. The molecules involved, such as glutamate and calcium ions, help stabilize these connections, reinforcing the circuits that define learning. Over time, repeated experiences deepen these patterns, making them easier to access. In essence, to remember is to reshape the brain, to alter its physical landscape in response to the world. Every thought, every moment of reflection, leaves its mark, weaving new patterns into the tapestry of the mind. Emotion, though often considered intangible, is as chemical as memory or movement. It begins deep within the brain, in regions such as the amygdala and hypothalamus, where sensory information is evaluated and translated into physiological responses. When danger approaches, for instance, the brain releases adrenaline and cortisol, preparing the body for action. The heart quickens, breathing deepens, and energy floods the muscles. When safety returns, other chemicals, serotonin, oxytocin, and endorphins restore calm and connection. These processes are ancient, inherited from the earliest animals that learned to respond to their surroundings. Yet in humans, they have evolved to encompass not just survival, but empathy, creativity, and love. The chemistry of emotion does more than regulate the body. It binds individuals to one another. Oxytocin, often called the bonding molecule, deepens trust and attachment between parents and children, friends and partners. Endorphins create a sense of comfort and well-being, easing pain and reinforcing social bonds. Even laughter and touch release cascades of neurotransmitters that strengthen connection. Through these molecular exchanges, the brain transforms social interaction into biochemical unity. It is as though our molecules recognize in others the same patterns they hold within themselves, bridging the space between minds through shared chemistry. Thought and emotion are not separate realms, but interwoven aspects of the same process. Every feeling gives rise to thought, and every thought carries emotional weight. When we recall a joyful memory, dopamine and serotonin flow. When we imagine loss or danger, cortisol and norepinephrine rise. The boundary between mind and body dissolves. What we think changes how we feel, and what we feel shapes how we think. This feedback loop, continuous and silent, defines our experience of being. It reveals that consciousness is not confined to the brain alone, but extends through the body, echoing in the heart, the breath, and even the gut, where networks of neurons mirror the patterns of the mind. Though the mechanisms of thought are material, their expression transcends the mechanical. Out of the exchange of ions and molecules arises imagination, the ability to create worlds that do not yet exist. When the brain recalls a memory or dreams of the future, it reactivates patterns of neural activity, blending fragments of experience into new forms. The neurotransmitters that once responded to reality now respond to the mind's own creation. In this way, the same chemistry that guides movement and emotion becomes the source of art, science, and philosophy. The molecules that shaped the earliest life now compose the poetry of human thought. Yet this delicate system is also vulnerable. Stress, deprivation, trauma can disrupt the balance of neurotransmitters, altering perception and mood. Chronic stress, for instance, keeps cortisol levels high, interfering with memory and suppressing serotonin production. Imbalances in dopamine or serotonin may lead to depression, anxiety, or addiction, conditions that remind us how dependent consciousness is on chemical harmony. Healing, in turn, often involves restoring that balance through rest, nourishment, connection, or medicine. The mind, like all living systems, seeks equilibrium. It heals not only through molecules but through meaning, through experiences that realign the chemistry of hope and calm. The mystery of thought lies not in its material components but in the unity they create. Billions of neurons fire in patterns, yet from their collective motion emerges awareness, the ability to observe, reflect, and understand. The mind becomes aware of itself, and through that awareness, it finds purpose. No single neuron knows the thought it helps to form, just as no molecule knows the emotion it transmits. Yet together, they produce the phenomenon we call consciousness. This emergence, the birth of mind from matter, is the quiet miracle of biochemistry, repeated every moment in every brain. In the grand continuum of life, the chemistry of thought connects us to all living beings. The same ions that carry signals in the human brain flow through the nerves of insects and the roots of plants. The molecules that create emotion in humans also guide behavior in other creatures. The dopamine that rewards learning in our minds also drives the songs of birds and the play of dolphins. Consciousness, in this sense, is not an invention, but a refinement, a deeper resonance of the same molecular symphony that animates all life. When you close your eyes and drift into thought, you are participating in this timeless process, the movement of atoms and energy arranged in the pattern of awareness. Each spark of understanding, each wave of feeling is both ancient and new, carrying forward the chemistry that began with the first living cell. Thought is life reflecting upon itself, the universe made conscious through the molecules that remember and respond. In the stillness of reflection, one can sense the gentle unity beneath it all, the heartbeat of biochemistry echoing in the pulse of the mind. Every neuron, Every chemical messenger, every fleeting emotion is part of a single, continuous flow, the living current that began in the oceans of the early earth and now hums quietly within your own brain. The chemistry of thought and emotion is, at its essence, the chemistry of being a tender bridge between matter and meaning, between the physical and the profound. And through it, life not only survives but awakens, feeling the wonder of its own existence. 19. The language of DNA, life's timeless script. In the deep silence of every living cell lies a language older than memory, written not in ink or sound, but in the sequence of molecules. This language is DNA deoxyribonucleic acid, the timeless script of life. Every organism that has ever lived, from the simplest microbe to the most complex human being, carries within it this molecular text, composed of 4 simple letters that together form an infinite library of instruction. It is a story written in the fabric of existence, a code that has guided the growth, behavior, and evolution of life for billions of years. To understand DNA is to understand not only biology, but also continuity, the unbroken thread that connects every living thing to the dawn of life itself. The structure of DNA is both elegant and profound. Each molecule is a double helix, 2 long strands twisted around one another like a spiral staircase. Along each strand are sequences of nucleotides, the fundamental units of this code. Each nucleotide contains a sugar, a phosphate, and one of 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. These bases pair with exquisite precision, adenine always with thymine, cytosine always with guanine, forming the rungs of the helix. This pairing is the secret to DNA's stability, its ability to replicate, and its power to preserve information across time. Within this molecular symmetry lies the quiet assurance that life, though fragile in form, is immortal in pattern. When a cell divides, it must copy its DNA, ensuring that each new cell inherits the same instructions. The process is both delicate and exact. Enzymes unwind the double helix, separating the 2 strands. Each strand then serves as a template for a new partner, as free nucleotides float in and align with their complements. The result is 2 identical helices, each containing 1 original strand and 1 newly formed. This method of replication, simple yet perfect, allows DNA to pass its message from generation to generation with astonishing fidelity. Mutations occur rarely, but when they do, they become the seeds of change, the subtle variations upon which evolution acts. The code of DNA is not just a static record, it is an active language. Specific sequences of bases form genes, each carrying the instructions for building a particular protein. Proteins, in turn, perform nearly every function in the cell. They act as enzymes, structural supports, messengers, and transporters. The process by which DNA gives rise to proteins is one of the most beautiful examples of molecular communication. First, the information stored in a gene is copied into a strand of messenger RNA, a process called transcription. The RNA, carrying the genetic message, travels out of the nucleus to a ribosome, where it is translated into a chain of amino acids. The order of these amino acids determines the shape and function of the resulting protein. From this flow of information, DNA to RNA to protein, arises the machinery of life itself. What makes this process so extraordinary is its universality. The same genetic code operates in every organism on Earth. A gene from a human cell can be placed into a bacterium, and the bacterium will read it and produce the same protein. This unity reveals a profound truth. All living things share a common origin. The language of DNA is not the invention of any single species, but the inheritance of all. Across 4 billion years of evolution, through the branching of species and the rise and fall of countless forms, This code has remained largely unchanged. It is a script so enduring that it binds the present to the past, and the simplest creature to the most complex. Yet DNA is more than a blueprint. It is a living narrative, capable of responding to the environment and adapting over time. The genes within a cell are not all active at once. Some remain silent. Others awaken only when needed, depending on signals from the surroundings. This regulation allows the same genetic material to give rise to many different cell types: muscle, nerve, blood, and bone, each expressing only the genes required for its role. Chemical modifications to DNA and its associated proteins, a field known as epigenetics, further refine this control. These changes do not alter the underlying sequence but affect how the code is read, providing a molecular memory of experience. Through such mechanisms, the environment leaves subtle imprints upon the genome, shaping not only individuals but, across generations, entire species. The resilience of DNA lies not only in its stability but also in its flexibility. It can repair itself when damaged, guided by enzymes that detect errors and restore the correct sequence. It can also recombine, exchanging segments with other strands to create diversity. This capacity for change underlies the adaptability of life, from the development of antibiotic resistance in bacteria to the evolution of new species. In every mutation, every rearrangement, the code experiments, exploring new possibilities while preserving its essential order. It is as though DNA is both writer and reader, both memory and imagination, eternally revising the text of life. Within the human genome lies approximately 3 billion base pairs, arranged in 23 pairs of chromosomes. Yet only a small fraction of this code directly encodes proteins. The rest, once dismissed as junk, is now understood to play vital roles in regulation, structure, and timing. Some sections act as switches, turning genes on or off. Others serve as scaffolds, organizing the architecture of chromosomes. Still others hold ancient fragments of viral DNA, remnants of encounters between our ancestors and the microscopic worlds that shape them. The genome, in this sense, is not a static manual but a living archive, a record of struggles, adaptations, and collaborations written in molecular form. Every human being carries within them this same code, yet each sequence is unique. Tiny variations in DNA, single nucleotide changes, insertions, or deletions distinguish one individual from another, influencing appearance, physiology, and even susceptibility to disease. Yet beneath these differences lies overwhelming similarity. More than 99% of human DNA is shared across all people, a reflection of our shared ancestry and unity as a species. Even more astonishing, humans share large portions of their DNA with other animals, about 98% with chimpanzees, 85% with mice, and even 60% with fruit flies. These shared sequences remind us that the boundaries between species are not walls but gentle transitions, branches from a common root. The study of DNA has illuminated not only the mechanics of life but also its continuity. Every cell in your body carries a copy of DNA that was passed down from your parents and theirs before them, in an unbroken chain, stretching back to the first living cell. Each replication, each generation, is another turn in that ancient spiral. The DNA in your cells is, in a very real sense, a living memory, a direct link to the earliest forms of life that once drifted in the primordial oceans. Through you, and through every living being, that original spark continues, unchanged in essence, though endlessly evolving in form. There is a quiet poetry in this molecular language. It has no author and no audience, yet it tells the story of everything that lives. Its letters are not written on pages but arranged in molecules, yet they carry meaning more profound than any human word. It is a language that speaks through structure, through replication and transformation, through the continuity of being. DNA is not simply the code of life, it is life's enduring voice, whispering through time, reminding every creature of its shared origin and its place in the unbroken chain of existence. To reflect upon the language of DNA is to sense the unity beneath all diversity. The same four letters have written every creature that walks, swims, or flies. The same spiral of information spins within every cell, connecting us to the distant past and the infinite future. When you breathe, think, or dream, you are enacting instructions written long before humanity existed, a script refined by eons of evolution, yet still unfolding, word by word, within you. The DNA within your body is a continuation of the same story that began when the first molecules learned to copy themselves under the ancient sun, and through you, its story continues a quiet, unending testament to life's persistence, written in the simplest, most beautiful code the universe has ever known. 20. The breath of life, oxygen, and the energy of existence. Every breath you take is a continuation of one of life's oldest rituals, the exchange between organism and atmosphere, between self and world. It is a rhythm so constant that it often goes unnoticed. Yet it is the very foundation of existence. From the moment you inhale, oxygen enters your body and begins a journey that connects you to every living thing on Earth. This single element, colorless and invisible, has shaped the history of life, driven the evolution of complex organisms, and sustains the energy that allows you to think, move, and dream. The breath is not merely a physiological act, but a reflection of the deep chemistry that binds all beings together in the quiet continuity of life. Oxygen, as an element, is both ancient and transformative. It was not always abundant on Earth. Billions of years ago, before the first plants and animals existed, the planet's atmosphere was dominated by gases like methane, ammonia, and carbon dioxide. The seas were rich in iron and sulfur, but devoid of free oxygen. Then, through the patient work of cyanobacteria, microscopic architects of transformation photosynthesis emerged. These early organisms learned to use sunlight to split water molecules, releasing oxygen as a byproduct. Slowly, over millions of years, oxygen began to accumulate in the oceans and atmosphere, forever changing the course of evolution. What was once a poisonous gas to ancient microbes became the essence of vitality for all aerobic life that followed. In the modern world, oxygen remains the unseen thread that links the biosphere. Every plant, through photosynthesis, converts sunlight into chemical energy, releasing oxygen as a gift to the air. Every animal, in turn, takes in that oxygen and uses it to unlock the energy stored in food. The two processes, photosynthesis and respiration, form a perfect cycle, one producing what the other consumes, sustaining the balance of life. When you breathe out carbon dioxide, it may rise into the sky and enter the leaves of a tree. Within those leaves, light will transform it once more into oxygen, which returns to you through your next breath. Thus, the simple act of breathing is part of a vast and ancient exchange, a quiet dialogue between life and the planet itself. Inside your body, oxygen's journey is both intricate and profound. When you inhale, air travels down your trachea, through branching bronchi, and into the delicate alveoli of your lungs, tiny sacs surrounded by capillaries thinner than a strand of hair. Here, oxygen diffuses across microscopic membranes into the blood, where it binds to hemoglobin, the iron-containing protein in red blood cells. Hemoglobin is a masterpiece of molecular design, capable of carrying 4 oxygen molecules at a time, holding them gently until they reach the tissues that need them. As the blood circulates, oxygen is released wherever it is required, diffusing into cells and entering the mitochondria, the powerhouses where energy is made. Within the mitochondria, oxygen takes on its most vital role, It becomes the final receiver of electrons in the process of cellular respiration. As glucose is broken down through glycolysis and the citric acid cycle, high-energy electrons are extracted and passed along the electron transport chain, a series of protein complexes embedded in the inner mitochondrial membrane. With each transfer, energy is released and used to pump protons across the membrane, creating a gradient that drives the synthesis of ATP, the universal energy currency of life. Oxygen waits at the end of this chain, accepting the electrons and combining with protons to form water. This simple reaction, quiet and invisible, sustains every heartbeat, every thought, every motion. Without oxygen, the chain would halt, and life's rhythm would stop within moments. The chemistry of respiration is a reminder of how intimately life depends on balance. Oxygen is both giver and taker, essential for energy, yet capable of destruction when uncontrolled. In its reactive forms, known as free radicals, oxygen can damage DNA, proteins, and membranes. To counter this, cells have evolved antioxidants, molecules that neutralize excess reactive oxygen species, preserving stability. Vitamin C, vitamin E, and enzymes like superoxide dismutase and catalase work continuously to maintain this equilibrium. This delicate management of oxygen reflects a broader truth of biochemistry, that life thrives not through abundance or scarcity, but through balance, the precise harmony between energy and restraint. Oxygen's influence extends beyond the cellular level to the very structure of the biosphere. The abundance of oxygen in the atmosphere allowed for the rise of multicellular life, for the development of complex organisms with high energy demands. Muscles, brains, and nervous systems all rely on rapid and efficient energy, release, something only possible through aerobic respiration. The growth of oxygen in Earth's atmosphere, known as the Great Oxidation Event, transformed evolution itself, leading to new ecosystems, and ultimately to the rich diversity of life we know today. Every living creature, from the smallest insect to the largest mammal, carries within it the legacy of that ancient transformation, a body shaped by the chemistry of air. Even the color of blood, the red that pulses through your veins, is a reflection of oxygen's bond. When oxygen binds to hemoglobin, it changes the structure of the molecule, altering the way it absorbs and reflects light. The bright red of arterial blood is the sign of life's energy in motion. The darker hue of venous blood is the sign of its return, depleted but ready to begin the cycle anew. This continuous circulation, intake, exchange, release mirrors the rhythm of the earth itself, with its own cycles of atmosphere, ocean, and light. In every breath, the same pattern repeats, the giving and receiving that defines existence. The connection between oxygen and consciousness is also profound. The brain, though only a small portion of the body's mass, consumes about 1/5 of the oxygen you inhale. Every neuron depends on oxygen to sustain the electrical activity that forms thought, perception, and awareness. When oxygen levels drop, consciousness fades. When it returns, awareness blossoms again. This link between breath and mind has long been recognized in human culture. Across traditions, the act of breathing is seen as sacred, the bridge between body and spirit, between the individual and the cosmos. In a biological sense, this symbolism is deeply true. The breath connects the inner chemistry of life to the outer rhythms of the earth. Oxygen's story is also the story of time. The air you breathe today contains molecules that have circulated for millions of years, molecules exhaled by ancient forests, by creatures long extinct, even by your own ancestors. Each breath is a reunion with the past, a communion with all who have lived before. The atoms of oxygen you inhale may once have been part of the lungs of a whale, the leaves of a fern, or the volcanic gases of early Earth. In this way, breathing is not merely an act of survival, but an affirmation of connection, a reminder that life, though fleeting in form, endures in substance and motion. The breath also unites all living beings in one shared rhythm. The oxygen released by a rainforest becomes the air inhaled by a mountain fox. The carbon dioxide exhaled by that fox nourishes the plants that feed insects and birds. Even the oceans participate in this exchange, as waves absorb gases from the air and release them again. The entire planet breathes a living, pulsing system that depends on the balance of air and life. When the balance is disturbed, as forests are cut and oceans warm, the planet's breath becomes strained, reminding us that our own well-being is inseparable from the health of the earth. To reflect upon oxygen is to recognize how fragile and miraculous life truly is. It is a reminder that existence depends not on strength or dominance, but on the quiet chemistry of connection. The same molecule that fuels your body also shapes the clouds, nourishes the seas, and flows through the veins of every creature that has ever lived. It is a bridge between the living and the lifeless, between the individual and the infinite. As you sit in stillness and breathe, you are part of this vast exchange, the same rhythm that began when the first photosynthetic organisms released oxygen into the sky. Every inhale is an acceptance of life's gift. Every exhale is a return of that gift to the world. The breath, in its simplicity, contains the entire story of existence. Energy drawn from the sun, transformed by the earth, carried through time, and given shape in the quiet chemistry of being. In every breath, life renews itself, a soft reminder that though the body may be transient, the rhythm it follows is eternal.